This is a platyfish, Xiphophilus maculatus, showing an anal fin graft, the gonopodium implanted into the side of the host. Actually, the graft was placed into this host before it has reached sexual maturity. And after the fish reaches sexual maturity, under the influence of androgenic hormone, the anal fin, the transplanted anal fin transformed into gonopodium. Next slide. It shows another platyfish with a transplanted anal fin transformed into gonopodium. Next slide. Could we have perhaps the lights off in the back? This is a platyfish which received a caudal fin graft. Notice the caudal fin graft is implanted into the side, and of course it is smaller than the natural caudal fin. This host was 6 weeks old when it received the graft. The graft came from a donor about 5 days old. The next slide. This shows us one of our inbred stocks of swordtails, which received, at the age of 2 weeks, a caudal fin graft. And as the fish reached sexual maturity 9 months later, under the influence of androgenic hormone, the grafted anal fin differentiated into the sword. Next slide. This shows one of the gynogenetic Physilia formosa with a heart graft sticking out on the side. Push the heart into a muscular pocket just below the skin. And as the host increases in size, the graft will also increase in size. And eventually will little bulge out through the body surface. Next slide. Shows another platyfish with a dorsal fin graft. Next. This shows one of our hybrids between two inbred stocks, but belong to different species, and they have a graft of melanoma on the side. Next slide. Oh, that's all. And this shows the histological picture of a fin graft, showing this here are the embedded fin rays and basal plates. Okay, thank you.
Coffees are waiting. We still have a formal discussant, and I don't want you to miss your lunches. So we are going to reach a compromise. I'm going to declare a 5-minute break so you can get yourself coffee, bring your coffee to your chairs, and we can hear our next speaker.
Well, Dr. Coleman said he had some difficulty with this topic. I must say I had a great deal of difficulty with this topic, and after I accepted Bill Hildeman's invitation to participate in this symposium, I sort of began to wonder what I'd gotten myself into. Because the fact is I know absolutely nothing about transplantation in fish, or even genetics in fish for that matter. My own research has been in transplantation in the mouse, which we don't get to until this afternoon, and even there with some subtleties of the genetic fine structure of the H2 region, which doesn't really have much direct relevance to this symposium. But I noticed that Dr. Coleman's paper was the only one whose main theme seemed to concern genetics. And I noted yesterday that the principal concern was with the phylogeny or evolution of the immune response, with not much reference to the evolution of the transplantation antigens, which is the other side of this coin, which I hope we won't forget about. So I decided that maybe I could function best here by simply reviewing, and it'll have to be very briefly, a few of the general genetic aspects of transplantation that may have some relevance here. Now, because my experience is with the mouse, where the best genetic evidence is available, most of my comments will be based on mammalian systems, with the mouse as the particular point of relevance, of reference. Now, first of all, the relevant genes involved in transplantation fall into two general categories. And these are the H genes, which control histocompatibility antigens, and the IR genes, which control the immune response. And both of these, of course, as I'm sure you know, have been defined in the mouse. Now, with regard to the H genes, Dr. Coleman pointed out the multiplicity of histocompatibility loci in various species of fish where it was possible for him to study these. And the estimates, based on the kinds of tests that he discussed, ranged from 3 to about 15 different loci in various species. Now, with a couple of exceptions, this is the rule also in most species where this can be studied. And in the mouse, the estimates from this kind of study go up as high as 30 loci. And in fact, thus far, some 22 loci have been defined in the mouse by straightforward genetic identification. Loci which control cellular antigens, not necessarily all strictly histocompatibility antigens. So it seems likely that the actual number of loci that control cellular antigens is going to be quite large. Now, the question has come up regarding the major locus in lower vertebrates. And it would certainly be very important to know whether fish and other lower forms have a major locus comparable to the strong loci defined in the mouse and the rat and so on. We tend to take it for granted that, at least in the higher vertebrate species, that such loci exist, but we really need a lot more information in the mammals and birds as well as in the lower vertebrates. And if we're interested in evolution, we have to know whether these loci or systems, strong systems defined in the various species and various classes, are genetically homologous. Now, without going into the details on it, I think the evidence is now quite good, based on a number of kinds of evidence, for homology of H2 and HLA, the human and the mouse systems. But we need to have similar kinds of information on a variety of species. Now, if the major locus doesn't occur in the lower vertebrates, why doesn't it occur? It may just be that these genes haven't evolved yet in these lower forms. But on the other hand, maybe the genetic homologs of the major loci occur in the lower forms, but there's something different about the mechanism of response so that they don't appear as strong. It's a possibility which Dr. Hildeman rather excluded this morning. Or perhaps the homologous genes are present, but they are invariant. There are not polymorphisms for these homologous genes in the lower forms, thus they can't be recognized through graft rejection. Or it's possible that the strong histocompatibility systems, which seem in general to consist of clusters of related genes, whose products are strongly antigenic, at least in part because of a cumulative effect. These clusters may, in some species, have been broken down through chromosomal rearrangement so that we don't find the cluster, we find components of the cluster on various chromosomes, and these are less strongly expressed as a result. Or on the other hand, it may be that it's necessary to get multiple gene duplications to generate these clusters of loci, genes which characterize the strong systems. And then in some species, the necessary degree of duplication just hasn't taken place. Well, this is all very speculative. The point is that, again, we need information on a variety of species and classes. Certainly, we should applaud Dr. Coleman and Dr. Hildeman and others who've made the effort to get this kind of information under difficult circumstances from species that aren't readily amenable to this kind of analysis. But the fact is that we still need to know a lot more about histocompatibility loci in various species. And still another aspect of the histocompatibility genes, important from the genetic point of view, is the biological function of the genes that control these antigens and the mechanisms that maintain multiple alleles, that maintain polymorphism at so many different loci. I won't take time to go into a lot of details. Again, there have been a lot of speculations. I think we might just point out that there's no reason to think that all of the histocompatibility loci have to fall into the same pattern, that they have to have the same function or similar, even similar chemical properties. It's conceivable that any gene which has something to do with a structure on the cell membrane is potentially a histocompatibility gene. But more specifically, it's been suggested, for example, that the H2 antigens are components of the cell membrane, that they're permeases or some cell-bound enzymes. They persist through long periods of cell culture, for example, which suggests that they may be rather critical to the physiology of the cell. It's been speculated that they may be involved in some surveillance mechanism against somatic cell variants. More recently, it's been shown that the H2 region, or at least genes closely associated with the H2 region, play a very important role in resistance or susceptibility to certain leukemia viruses, and also a role in the level of the antibody response to certain synthetic antigens. So another role. And just a couple of days ago at the histocompatibility workshop, Dr. Walford presented some very intriguing data suggesting that the HLA antigens may play a similar role in susceptibility to one form of leukemia. So I have the feeling that the pursuit of this kind of observation may lead us to some rather important understanding of the function of these antigens. Now, with respect to the IR genes, back to the other side of the coin, Dr. Coleman discussed some conditions under which variability in the both genetic and non-genetic variability in the immune response may be observed. And one other aspect here is the immune response genes, the genetic the genetic constitution of the host, which certainly in at least non-inbred animals must have a very important role in the type of immune response that's observed. There are a number of cases now in which single gene control, single gene differences have been shown to play a role in the level of the antibody response to specific antigens. And several of these cases involve histocompatibility loci. There's the case which I alluded to earlier in which genes responsible for response to synthetic antigens are closely associated with the complex H2 region. McDevitt has shown this with synthetic polypeptide antigens. Rathbun and Hildeman have shown this for the hapten TNP in the mouse. And there are also some other, so far unpublished, indications of H2 involvement in response to other antigens. It's also possible, although it hasn't yet been entirely verified, that another IR gene in the mouse, the IR2 gene, which controls antibody response to a cellular antigen, a red cell antigen, may be associated with the H3 histocompatibility locus. So here we have a situation in which one histocompatibility locus may be involved in the antibody response to a cellular antigen determined at another locus. Well, at any rate, it's pretty clearly established that there are single genes that can control the antibody response to specific antigens. And it seems quite possible that similar genes controlling the cellular response may also eventually be demonstrated. And then, of course, there are all the structural genes for immunoglobulin chains which have been demonstrated, and surely these play a role in the response as well. So the point is that we need to remember that the strength of the immune response is a function not only of the properties of the antigen, which we should remember, but also the genetic makeup of the host. And here I would emphasize the genetic makeup of the host. Now finally, just one general observation about the genetic aspects of transplantation. We're interested in the evolution of the transplantation reaction. And I think we should keep in mind that evolution proceeds through mutational changes in specific genes. And certainly one important approach to the understanding both of the evolution of the transplantation antigens and the evolution of the immune response to these antigens is a better definition of the specific genes involved, and that this needs to be done at a number of phylogenetic levels. Thank you.
Unless someone has a very burning question that must be answered on the spot, we shall proceed to the next paper because of time shortage. And it's Transplantation, Immunity of Apada and Urodela by Nicholas Cohen of Microbiology, University of Rochester, School of Medicine and Dentistry.
When I was a postdoctoral fellow with Dr. Hildeman a few years ago, I remember one laboratory group session very vividly. And in this group discussion, Dr. Hildeman was advising us as to what we should do when we present papers. And one of his strong admonitions and cautions was that we make sure we listen to the preceding speakers. Obviously, I was here to come to this meeting. I have heard the preceding speakers, and I'm very pleased about what they've said, because I think I'm being set up to give my paper. What has been said before me serves as a magnificent introduction to many of the points I'd like to cover. Now, as we've heard today, one obvious goal of a comparative immunologist is simply to catalog a variety of immune responses of a given vertebrate species, and to then compare these immune responses in a phylogenetic fashion. Now, it's the hope of some of us here that the very same data that allow this cataloging to occur may also reveal new animal models with which problems of common interest to all immunologists, and also of common immunologic phenomenon may be revealed. And what I'd like to do this morning is to catalog the immune responses, the transplantation biology of urodele and apodont amphibians, two orders of amphibians, and then indicate how these may well be a model system of weak histoincompatibilities in natural populations, paying particular emphasis on immunologic memory. May I have the first small slide, please? Now, this was one point that I'd like to make as a discussion of Dr. Coleman's presentation. This is the salamander that we use. This implant here is an allogeneic heart. We find identical data to that of Coleman's, where hearts survive quite longer than simultaneously transplanted skin allografts. In fact, we find in normal non-transplanted animals just receiving a heart implant, we find about 80% survival, 80, 100, 200, 300 days post-transplantation. Now, this particular animal, you can see white spots here. This animal, can you focus that, please? This animal, these are signs of where grafts have been placed and have been rejected. Thank you. In other words, this animal had been pre-treated with 4 or 5 repeat grafts from the original donor, that same donor that supplied the heart. And this heart is continually beating. This is at 100 days post-implantation. We'll get back to this at the very end of my talk. May I have the next small slide, please? Again, the newt, Dimictylus viridescens. You can see here the model we use for transplantation studies, either implantation, in this case a liver, which produces a profound immunosuppressive effect against skin transplants. Here, a skin allograft placed from another animal from the dorsal flank containing many melanophores onto the throat. And we use the pigment cells, as do other comparative transplanters, as an index of graft viability. May I have the first large slide? Now, there's no question that salamanders can reject skin grafts. Here we see an allogeneic transplant, extreme vasodilation, small hemorrhaging, seen histologically, lymphocytic infiltration. Now again, of this flank graft placed on the throat, hemorrhage areas where the arrows are pointing, and increasing infiltration of small lymphocytes with still viable skin glands. Now these photographs were made at 24 days and 28 days post-transplantation at 25 degrees centigrade, and all the studies I'm going to talk about are animals maintained under fairly decent laboratory conditions at 25 degrees centigrade. Next slide, please. And here's an example of complete pigment cell destruction, 36 days post-transplantation, the absence of any graft structure, and actually the lymphocytes have already been leaving the transplant area. Next large slide, please. Now, these two transplants are also allografts. These small black dots are pigment cells. These two allografts are both fully viable. There's no lymphocytic infiltration. These photographs were taken 200 days post-transplantation at the same temperature. In other words, this is somewhat of a contradiction of Medawar's earlier statement that you saw in the introductory remarks that Dr. Cooper made. The salamander, in fact, is not resolutely intolerant of skin grafts. The salamander presents us with a model of chronic rejection, 36-day typical median survival times, with roughly 10% of the thousands of grafts we've performed on these animals exhibiting extremely prolonged survival such as these. Next slide, please. Now, although urideals have not been studied in terms of the number of species to the extent that hyateliasts have, transplantation biological responses have been observed in about 15 species, representing 9 families and 9 genera and 4 families. And in a comparative study, we were determining the first and second set median survival times at 25 degrees of a variety of different salamander species. And one thing becomes apparent. Chronic rejection reactions are typical of outbreeding urideal amphibians. There's been one study on the apodine amphibians, another order, where chronic rejection is also characteristic. Also, the second set survival times, median values now exhibit accelerated rejection, and the broad first set and first set survival ranges with possible prolonged indefinite survival are indicated here. Again, typical of a chronic rejecting system, be it H2 compatible mice or salamanders. Next slide, please. Now, those previous data were for allogeneic combinations. Similar things have been observed in xenogeneic combinations. And here we have intergeneric and interfamilial transplants, reciprocal transplants, seen over here. And by and large, the median survival times are also chronic. There are some indications of subacute rejection. There are also indications of indefinite survival. When I left my postdoc with Dr. Hildeman, he still had this animal. Now, the obvious question is, how do we account for this kind of chronic rejection? And certainly Dr. Schreffler posed a very important question. May I have the next slide, please? Is this a function of an anatomically impaired immune response capacity? Well, you can see that category is not indicated on this slide. It's not indicated simply because, under certain conditions, we have observed vigorous alloimmune responses. And by vigorous, I mean first set rejection at 7, 8, 9, 10 days, comparable to fish or mouse. The reasons, the explanations that I'd like to offer to you today to explain the chronic rejection are as follows. And Dr. Schreffler has again indicated several of these in his talk. An absence of a major H2 type of complex histocompatibility locus, cumulative interactions of weak transplantation antigens, antigen sharing, and a genetically imposed limitation on the immune response capacity, the IR response, the IR region, the chromosome. Now, what evidence do we have? These are outbreeding animals, and by this we're hampered, unlike the work with fish, by the lack of inbred strains, or rigorously defined inbred strains. Now, I indicated earlier that subacute and acute rejection responses have been observed, but they've been observed in 2 highly defined conditions. The first condition, where we observed the subacute median survival times, and by subacute I mean rejections of about 22 days, was in a population of salamanders collected from one pond. And we repeatedly went into this population at different times of the year, pulled out animals, and repeatedly got the same subacute median survival times, in contradistinction to the more chronic median survival times seen typically in other populations of the same species. Next slide, please. So we have population A, typical 22-day median survival times, and population B with typical chronic median survival times. Now, in deference to the people who were at the HLA workshop, let me present some quasi-genetic analysis indicating why we believe there's an absence of a major histocompatibility locus in these species, at least one of the reasons, and why we believe that chronic rejection is in fact due to weak histoincompatibilities. If we reason that population B rejected grafts slowly, simply because it lacked strong transplantation antigens, and that population A animals rejected intra-population allografts because they possessed strong transplantation antigens, then grafts from population A on animals from population B should be rejected in a subacute fashion. This was not the case. Chronic rejection was the typical mode. Likewise, if we believe that population B animals possessed strong transplantation antigens but shared the same transplantation antigens with each other, that would also explain the chronic rejection. But then grafts from population B with a given set of strong antigens on population A with an obviously diverse set of transplantation antigens which would be strong would be rejected in a subacute fashion. This did not happen. We found chronic rejection. Now, there are a variety of other reasons that we believe we're not dealing with strong antigens. But how then can we explain the subacute rejection? We simply, in this case, extrapolate from the mammalian literature where we know that the cumulative interaction of weak transplantation antigens can readily lead to acute and subacute rejections. Lights for a second, please. Well, there's one other area that we haven't covered in connection with the basis of chronic rejection, and that is antigen sharing. And we studied antigen sharing by the use of the third-party test. Simply transplant a graft from animal A to animal B, get a first set rejection time, transplant a repeat graft from that same donor to the original recipient, and get the typical accelerated second set reaction. And what happens when you transplant, instead of the repeat graft from animal A, a graft from a third party, animal C? And if the graft C is rejected in an accelerated fashion, obviously animal A and C share effective transplantation antigens. And we've studied one population in this fashion, and we found a very high incidence of antigen sharing, something, of course, that would also be conducive to chronic rejection reactions. The transplantation antigen sharing has not been exhaustively studied, but an experiment by Davis using another species of salamander, the salamander received 4 separate transplants, each transplant coming from a different donor, and these transplants were placed at the same time. And after the rejection of these 4 individual grafts from 4 donors, that animal was sensitized to any and all 5th grafts. May I have the lights off, please, in the next slide? Now, the last point that I'd like to indicate in connection with the genetics of transplantation immunity and transplantation immune responses is what may be analogous to the IR locus. And this is simply a situation where we also found subacute rejection. And the second situation where we've routinely seen subacute and acute rejection responses has been in one salamander subspecies. Now, this is the Dimictulus viridescens dorsalis subspecies, and the one I've been talking about previously is Dimictulus viridescens viridescens. Now, we've collected dorsalis from all over a given state, and in fact from 2 states. These are not animals from one population. And what we find is that, in fact, rejection is routinely subacute. Median survival times ranging from about 19 to about 23 days. And the viridescens, where we have chronic rejections, median survival times from about 36 to about 42 days. Well, how can we explain the subacute rejection here? There are 2 possibilities. Possibly in the speciation of this subspecies dorsalis, there was a mutation of the histocompatibility locus involving new alloantigenic specificities, which would be relatively strong. The other possibility is that these animals are in fact better responders than the other subspecies viridescens. And the way we simply tested for this was to do inter-subspecific transplantation. And what we found was consistent with the fact that dorsalis is a better responder than viridescens. Because dorsalis not only rejected dorsalis allografts in a subacute fashion, but it also rejected viridescens allografts in a subacute fashion. And likewise, viridescens not only rejected viridescens allografts in the chronic fashion, but it too rejected dorsalis allografts in the chronic fashion. Now, obviously, in this kind of genetic system, there's a lot of work to be done. And of course, one question is, do dorsalis and viridescens, the 2 subspecies involved, also share transplantation antigens? Let's take a mental pause for a second and think about the organs involved in the transplantation alloimmune response in Urodela. Now, the most beautiful evidence that the spleen, liver, and perhaps the thymus are involved in mediating transplantation immunity comes from the work of Murakawa, a Japanese worker. May I have the next slide, please? And what Murakawa has done is to simply use as a reference animal an irradiated salamander. Now, both he and I have demonstrated that 450 and 500 Roentgens prevents this animal from mounting a transplantation immune response. So what Murakawa has done is, prior to irradiating this animal, removed spleen, liver, or thymus. And after irradiation, the spleen, liver, or thymus was replaced as an autograft back into the original recipient. And now this animal was immunologically reconstructed. He could reject transplants, as you can see over here. Next slide, please. Now, Murakawa pushed this type of experiment one step further. Again, the reference Synops pyrogaster irradiated, and this time this animal received an allogeneic spleen. Now, he did succumb to some extent to homologous disease, but note that his immune response had been restored. He could reject a third-party graft, but he did not reject skin grafts from the original spleen donor. Next slide, please. In a more complex series of experiments, again, using an irradiated animal and reconstructing this animal in the following way. First, B animal received a skin allograft from A. So now B carries a sensitized spleen, a spleen sensitized against A's transplantation antigens. Now this B spleen goes into this irradiated animal. This irradiated animal now receives 2 skin grafts. He receives a skin graft from a D animal, and he receives a skin graft from the A animal, the animal that initially supplied the sensitizing antigens. Now, the irradiated animal rejects A's graft in an accelerated fashion and rejects D's graft in a typical first set response. Now, these studies were performed with spleen, and to a lesser extent, liver carries out the same function. There are a couple of other areas of the transplantation biology I'd like to cover very briefly, if I could have the lights, please. The first thing I'd like to indicate is what is known about the development, the immunologic maturation of the transplantation immune response. Now, the transplantation alloimmune response, at least in 1 or 2 species that have been studied, matures quite late in larval development. That is to say, if you take an animal, it's 30 days post-hatching from his jelly capsule, and you skin allograft him with skin from non-siblings but of the same developmental age, he doesn't reject those grafts until much, much later than a control would. In fact, by about 60 or 90 days, that is about the time, about 3 months post-hatching, when the alloimmune response reaches control levels of reactivity. And as you might expect, if you take a 30-day-old larvae and thymectomize it at that time, and then transplant this animal when his control siblings are immunologically mature, this thymectomized animal does in fact not reject the skin allografts. By thymectomy as late as 50 days post-hatching, you've abrogated alloimmune responsiveness to transplantation antigens. One other point that I'd like to consider in the immune parameters that we measure transplantation phenomenon in, I'd like to talk about immunological tolerance. Now, tolerance has been produced in both embryos, larvae, and adult forms of salamanders with relative ease. In embryos, namely tailbud stages, the French group, Houillon, Charmagne, and Plumfiere, have produced tolerance simply by parabiosing embryos together in the tailbud stage. And these parabionts grow up and they metamorphose and they're perfectly happy. Tolerance has also been produced by reciprocal grafts of embryonic ectoderm made at the tailbud stage. The animals grow up, they're very happy, and the skin grafts, the skin is differentiated from the ectoderm, and it persists. Now, the length at which it persists depends upon the genetic factors involved. Some of the combinations were xenogeneic combinations. Even in this situation, the grafts survived. In some cases, they were eventually destroyed. Tolerance has also been produced in embryos simply by producing head-body chimeras. In other words, the anterior half of a tailbud stage of one genus has been elegantly but simply fused to the posterior half of another species. This odd-looking creature that develops is a chimera, and it's, again, maintained. It grows and lives and feeds. It's quite happy. Now, we've induced immunologic tolerance in larvae simply by taking a larval form which is immunologically incompetent at 20 days post-hatching, reciprocally exchanged tail-tip grafts. These tail-tip grafts survive for 100 days. This is our cutoff point. And in the few instances where we reciprocally exchange skin grafts between the original donor and host, these skin grafts also survive for 100 days. Tolerance in the adult salamander has been induced in two ways. The first, again by Murakawa, was to produce tail-to-tail parabionts in the adult animal, something that is no mean feat. And he found that although some of the animals succumbed to what he called parabiotic disease, that in the animals that survived, these animals were tolerant of skin allografts exchanged between the parabionts. In fact, they were not totally immunologic paralyzed because they were able to reject third-party grafts. Now, Murakawa also found that the degree of immunologic tolerance was dependent upon how long the parabionts were joined in cross-circulation. If they were separated after a long period of time, 100 days, they were tolerant. But if they were separated after about 30 days, they were not tolerant. In fact, they were sensitized to each other's skin allografts, implying there was a transition between immunity and tolerance. And we've also induced immunologic tolerance in adults simply by implanting larval tissue into the throat pouch of one of these salamanders. The throat pouch is not a privileged site. We can do this intraperitoneally also. But embryonic and larval tissue implanted into an adult xenogenic or allogeneic recipient continues its differentiations, its growth, and forms a large prodigious mass which looks like a teratoma in the sense that it consists of widely diversified tissues such as heart, skin, muscle, kidney, nervous tissue, etc. And these teratoma-like structures persist indefinitely. We have still some in our lab that have been about 2 years old. In fact, the fetal tissue, or the larval tissue, is tolerogenic, has been demonstrated by Dr. Delaney, who has some inbred strains of axolotls. And he did a similar kind of experiment and took the recipient bearing this large growth, transplanted skin from an individual of the same donor strain, and the skin graft also survived. Now, in the remaining time left to me, I'd like to discuss, may I have the next slide, please? Let me briefly summarize then some transplant phenomena common to H2-compatible mice and outbreeding irideals. What I'm going to try to indicate here is that both are chronically rejecting systems, and that both these phenomena and the ones in mice, which are the same, are characteristic of weak histoincompatibility interactions. Certainly, graft rejection is chronic with a broad range of survival time. We in salamanders see periodic rejection crises followed by either gradual destruction or long survival. Gradual infiltration with small lymphocytes is also seen. Relatively mild, if at all, graft versus host reactions, mild parabiotic reactions, prolonged survival of fetal tissue, ease of tolerance induction, and relatively long post-transplantation period prior to host sensitization. Now, what I'd like to do is to consider immunologic memory in this weak histoincompatibility interactions when we characterize the irideal amphibian. May I have the next slide, please? As you may well know, in H2 incompatible mice, a repeat transplant, a second set graft, is destroyed just about as rapidly as a third set graft. In other words, repeat immunization does not quantitatively alter the rapidity of repeat graft rejection. It may in fact qualitatively alter it in terms of the numbers of white grafts that you see after repeated immunization. This does not seem to be the case when we're dealing with weak histoincompatibilities. We have the median survival times of first, second, third, fourth, and fifth set grafts. And we're talking about median survival times where the number of animals involved were 51. And each host received first through fifth set graft. Each graft was placed following the rejection of the previous set. And each graft, of course, came from the original donor. And what you see here is the accelerated median second set values and a significantly accelerated third set. Now, the third set seems to represent, in the median value, the maximum response that animal is capable of eliciting against the antigens which challenged it. But the median survival times are somewhat misleading. They're misleading in the sense that they only reveal one kind of immunologic memory class, the accelerated second set and the accelerated third set. This is but one of three different kinds of anamnestic response categories that we've observed. May I have the next slide, please? Now here we see 6 of those 51 hosts. 2 of these hosts represent one type of memory response, and I've selected these hosts on the basis of the fact that they've rejected their first set grafts roughly about the same time. And I think you're going to see what we were talking about yesterday in concern with prolonged survival of second set transplants. This is the memory class that was generated the median survival times. The yes second set, yes third set response. Accelerated, 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 accelerated. There's also a memory class which is more comparable to what one sees in H2 incompatible mice with a second set but no third set. The second set then represents the maximal host response, and it's the minimal graft survival time. The minimal graft survival time generated by one transplant is the same as that generated by 2 transplants. Now here's the strange third anamnestic response, where there's no second set and there's a yes third set. And in this particular group, you see the second set survivals are somewhat prolonged. Note there is indeed a third set response. Now, may I have the next slide, please? Is there any distribution pattern of these 3 different kinds of memory responses? Indeed, there is. We're talking about first set survival times, which range from 20 days to 155 days. And the distribution of this no second set, yes third set was about 19%. Yes second set, yes third set, almost half. And the yes second set, no third set, about 35%. Now this distribution, this overall distribution, is very dependent on the rapidity of first set rejection. In fact, with increasing chronicity of first set rejection, this kind of response just about disappears. Only 6% of the hosts show this lack of a second set response in this particular range group where first sets were rejected between 42 and 155 days. Each range group represents about the same number of animals, 15 to 20 recipients. Now this category is most common when we're dealing with the most vigorous kinds of first set rejections, 20 to 29 days. We find 30% of the hosts rejected their grafts in that fashion. What is very strange and puzzling is 45% rejected the grafts with a full second set, a full-blown second set response, the maximum response, and no third set. To me, this obviously indicates a wide degree of antigenic diversity, and certainly a wide degree of what Dr. Hilderman has referred to as the genetic library or genetic repertoire of the recipient. We obviously have 3 entirely different kinds of memory responses given the same first set survival times. I'd like to bring this point home in the next slide, please. Now, this is a breakup on the individual survival times on hosts 1 through 20, those hosts that rejected their first sets from 20 to 29 days. And these are the actual values. Here's the no second set, yes third set response, and these are the mean values. And what's interesting here is, in fact, we're not dealing with prolonged survival. We're not dealing with significantly accelerated rejection. We're dealing with just about the same first set survival times, 2020, 2018, 2118. These are very similar survival times. And what I'd like to suggest as an explanation, not as an explanation, but as a point of conjecture, is that first, immunologic enhancement may be involved in the situations where we find this kind of response, or perhaps better to say that immunologic enhancement might be found in the situations where we find a prolonged second set survival. Now, the prolonged second set survivals occurred when first set rejection was between 35 and 155 days. It did not occur here. We may have 2 factors involved, again reflecting antigenic diversity. Some antigens and antigenic combinations may reveal an enhancing antibody, others may not. But forgetting about enhancing antibody this time, it's obvious in this situation where we did not get a second set response, but we did get a third set response, that we did get a first set response also. So obviously these animals were rejecting their first set grafts, but not demonstrating immunologic memory. And as I indicated, perhaps the events and stimulations leading to the first set rejection may be very different from those leading to anamnesis. Can I have the next slide, please? I'd like to summarize and perhaps it may be too premature to refer to these as additional properties of weak incompatibilities. Perhaps talk about this additional properties associated with chronic rejections. Certainly, the data with salamanders reveal that weak alloantigens are also weak xenoantigens. And these kinds of studies obviously cannot be done in a rat-mouse system because you cannot have congenic partners, one being a mouse and one being a rat. But since we feel we're dealing with non-strong transplantation antigens, the xenografts are rejected in this chronic fashion also. We have data that immunity conferred across the weak barriers, at least in the salamanders, by skin grafts, not by dissociated cells, is long-lasting. May I have the next slide, please? And I'll finish up in a second. What we did is simply to divide our animals into 4 groups based on the interval between the rejection of the first set and the survival and the transplantation of the second, 10-day, 30-day, 60-day, and 3 months. And we found the first set median survival times were very close to each other, and the second set survival times were very close to each other. And I must add that all 3 memory responses occurred in about the same distribution, regardless of the interval between rejection of first set and transplantation of second set. Last slide, please. This last slide summarizes what I've been saying and continues that summary. Animals hyperimmunized by multiple skin grafts will show increasingly curtailed survival endpoints until the minimal survival point, which is a reflection of the maximal host response capable of being generated against the transplantation antigens, is reached. Second set grafts may survive as long as or longer than first set grafts from the same donor. In such combinations, third set grafts are often more rapidly rejected. Although different antigens may result in the same first set survival, the state of immunity, they can confer, again, an indication of tremendous antigenic diversity in an outbreeding system. In this case, the salamander, perhaps in another case, people. Weak antigens may not immunize hosts as rapidly as strong antigens. I don't have time to present the data for this. A very intriguing possibility, which we've seen with fish, which we've seen with rats, which we've seen with salamanders, with the same weak histocompatibility differences, organs like heart and kidney may survive better than skin. And we can add ovary and various endocrine organs to this category also. And a point that we'll come back to at the end of this meeting, when again I would like to echo some of the things that Dr. Schreffler said about the evolution of transplantation antigens, as well as the evolution of the immune response against these antigens, is that weak histoincompatibility reactions characterize the response of most primitive vertebrates studied. Thank you.
Time is late, but we must hear from the formal discussant, Dr. Horton of Tulane University. However, the informal discussion from the floor will be entertained this afternoon during general discussion. Dr. Horton.
Dr. Siegel, ladies and gentlemen, most of the data on transplantation immunity in the Anura has come from studies on advanced representatives of this order, and these animals show acute alloimmune responses in contrast to the chronic rejection phenomena that Dr. Cohen has been talking about in urodeals and apodens. I have been working on the ontogeny of the immune system in the clawed toad, Xenopus levis, and I'm going to compare the immune response of this animal with that occurring in other amphibians. Now, morphological and karyological data suggest that Xenopus is a primitive Anuran representative. And our investigations in Hull in England on the immune system in this species are in accord with this suggestion. Thus, both the larva and the adult lack certain lymphoid organs, namely the lymph glands of the larva and lymph nodes in the adult, that are found in more advanced Anurans. And compared with the latter, the adult shows deficiencies in its immune response to injections of bovine serum albumin, which may be associated with its lack of lymph nodes. My own transplantation studies in the larva have shown that the immune response to first set skin allografts includes a lymphocytic invasion of the graft. This invasion is correlated with the state of development of the lymphoid organs of the host. If these organs are immature at the time of application of the graft, the invasion is delayed but eventually occurs when the host reaches a more advanced larval stage. The ability of the larva to invade a graft with lymphocytes is correlated with the lymphoid maturation of its thymus, this organ being of prime importance in establishing the immune system, as it is in both urodele and other inuran amphibians. Certainly in Xenopus, it seems that a relatively early exposure of the larva to a foreign antigen does not necessarily preclude the possibility of an immune response to that antigen as the animal matures. And thus this work is possibly in accord with Dr. Volpe's work on Rana pipiens, which he will be speaking about this afternoon. I should just mention at this point that Dr. Cohen quoted some work done in France, and he didn't mention that to an FEA showed that the transplantation of ectoderm between very early urodele larvae in some instances can be rejected in the late larvae. In other words, tolerance isn't necessarily induced by early larval allograft allografting in the urodele. Recent electron microscopic evidence on Xenopus allografts showing the resemblance of the invading lymphoid cells to the lymphocytes in the thymic cortex reinforces findings from light microscopy that the lymphocyte plays a vital role in allograft destruction in amphibians, as of course is the case from hagfishes upwards. And here I have one or two slides just to show. Could I have the lights out, please? This is just a light microscopic view of, through the cortical region of the thymus of a fairly late Xenopus larva, showing lymphocytes. You can see the nuclei standing out very clearly here, the lack of cytoplasmic staining with this particular stain used, and the denseness of the chromatin, and in some instances, here for instance, and here, the nuclear membrane stands out very clearly. Next slide, please. This is a section under a larval skin allograft placed on, again, a mature Xenopus larva. The graft would be up there, the epidermal region. You can just see the cutis or basement lamella here. This is a blood capillary underneath the larval skin allograft, packed mainly with lymphocytes. There are also lymphocytes near this blood capillary, extravascular lymphocytes in this area. This is a fairly early stage of rejection in Xenopus. Notice the similarity of these cells to those seen in the thymic cortex. Next slide, please. This is a low-power electron micrograph to show through the thymic cortex of a mature Xenopus larva, showing the appearance of lymphocytes in that region. You can see the high nuclear cytoplasmic ratio. There's the nucleus, cytoplasm, a typical feature of lymphocytes. What is not a typical feature of, say, mammalian lymphocytes in the thymic cortex is these pseudopodial-like outgrowths of the cytoplasm. The chromatin is light and dark chromatin is lumped, and you can see that near the nuclear envelope in this lymphocyte here, that there is a dark band which is the dense chromatin. In the cytoplasm, although it can't be clearly seen in this picture, are numerous mitochondria, and the density of the cytoplasm in this micrograph is due to the fact that the cytoplasm is packed with free ribosomes. Also note the tiny little vacuoles present in the cytoplasm. Next slide, please. This is an electron micrograph of the basal lamellar region, or cutis, as I showed you in the light microscopy slide previous, showing a lymphocyte passing through, or at least I believe is a lymphocyte, in the basement lamella. Notice the similarity in structure of this cell to the lymphocytes that I've shown you in the thymic cortex. The high nuclear cytoplasmic ratio, the density of the cytoplasm with the stain used due to the masses of free ribosomes that are found in this area. Also, the tiny vacuoles and pseudopodia. You can see there's been some disruption in the basement lamella in this region. And finally, next slide, please. This is a picture of a lymphocyte actually in the epidermal region of the allograft, above the basement lamella. Again, here in this picture, you can see quite clearly the mitochondria, the little vacuoles, and the little dark dots that it shows up here, which are the ribosomes. It's interesting that these cells in larval allografts at this stage are generally found between the cells in the intercellular spaces, which is similar to findings on mammalian work. Also, there is a close proximity of the plasma membranes of the epidermal cell there and the lymphocyte in places, again resembling some findings that have been found on mammalian allografts. Could I have the lights, please? Experiments on young post-metamorphic toads, which I'm going to call toadlets, have revealed that 75% of these reject first-set skin allografts from non-siblings in about 3 weeks at 23 degrees centigrade. Grafts on the other 25% were never entirely rejected within the course of the experiment, which was about 4 months, although all of these grafts showed some signs of necrosis, such as enlarged superficial capillaries and breakdown of the pigment at the edges of the graft in some areas. This latter type of response to skin allografts has been previously reported in work on urodeals. Dr. Cohen has observed that certain first-set grafts on Necturus showed signs of rejection, but with no additional degenerative phenomena. He has also observed that several first-set skin allografts on Dimectalis viridescens remained in place for longer than the normal chronic rejection period. In Charlemagne's laboratory in Paris, it has been recorded that necrosis of some first-set grafts on the urodale plurideles was hardly visible, and these workers surmised that the graft tissues were gradually replaced by those of the host, as may well occur in the present experiments on Xenopus. The French workers ascribed the outcome of their results to the close genetic affinities of the animals in their colony, which is also one possible explanation of the chronic reactions seen in my experiments. An alternative explanation is that 25% of the animals had a non-competent immune system. This seems unlikely, however, for although these animals did not destroy second-set grafts from the same donors as the first-set grafts, they rejected third-party grafts within 3 weeks. Toadlets which had rejected their first-set grafts in normal fashion generally rejected second-set grafts in an accelerated fashion, although a true anamnestic reaction was seen in only one animal. The outcome of the second-set reaction was shown not to be affected by variations in the time interval between application of first and second-set grafts. In my experiments, second-set grafts became revascularized prior to rejection. Now, an adequate blood supply is apparently not established to second-set grafts on larval leopard frogs, and Dr. Hildeman has described that recirculation to second-set grafts in larval bullfrogs rarely occurs. A white graft reaction has been observed in some second-set skin allografts on the Urodele plurideles. However, Dr. Cohen has shown that revascularization of second-set grafts often occurs in the newt. Now, in mammals, the balance between accelerated rejection reactions, in which revascularization of second-set grafts occurs, and the white graft reaction, where there is no revascularization of second-set grafts, the balance has been shown to be a very delicate one. And thus the differences found in respect of second-set grafts in my experiments and those described by John Simnett of England, in which revascularization in his experiments of second-set allografts on Xenopus toadlets did not occur, are most likely attributable to the greater genetic diversity of the toads he used, resulting in the more severe immunological reactions in his experiments. In my experiments, some sensitization to third-party grafts occurred, which tends to confirm suspicions that an appreciable amount of antigen sharing existed among the non-siblings used. This is also suggested by the fact that toads of the whole colony reacted more vigorously to skin allografts from an outside stock than to grafts from members within the colony. It's interesting to note here that one toadlet, having rejected its first set graft in normal fashion, did not reject its second set graft from the same donor as the first set graft. In his paper, Dr. Cohen has shown the occurrence of this phenomenon in urodeles, where antigenic sharing between donor and host is known to occur. The application of second set grafts to toadlets that had previously received and responded, in fact appear to have rejected the first set grafts in larval life, has given some rather unexpected, but I believe very interesting results concerning the immunological status of these animals. Thus, partial tolerance is achieved when first set grafts are of adult skin and an unchanged, i.e. first set immune reaction occurs after larval skin first set grafting. An accelerated immune reaction to second set grafts was never induced in these experiments, in contrast to similar work by Dr. Volpe on the leopard frog, in which rejection of allografted tissue in larval life results in immunity in the adult. I feel that further studies on Xenopus are required here to investigate these findings, as they may yield interesting information on the mechanism of immunological tolerance and immunity in amphibians. In conclusion, I believe that these experiments indicate that the immune system, and by the immune system I mean not only the transplantation response, but also the lymphoid organs that are present and the humoral antibody response, that I believe that this immune system in Xenopus is of a lower order than that seen in the more advanced aneurin representatives, such as the Rhinidae and the Buffonidae, and that certain features of the alloimmune response in this species resemble the chronic rejection phenomena that Dr. Cohen has been describing in urodeles and that Dr. Kuba has observed in apodon amphibians. Thank you.
Well, the time has passed.
Thank you, Dr. Storman. The studies that I will discuss were not designed initially to demonstrate transplantation immunity. I am one of those classicists that Dr. Storman has been referring to. The investigation began as a problem in developmental genetics, but became unavoidably a problem in immunology. I would like to share with you the stepwise development of the investigation. May I have the first slide, please? As seen in the first slide, The experimental animal is the familiar common spotted leopard frog, Rhonopippians, widely used in classrooms and in research laboratories. The next slide shows a conspicuous pigment variant of the common leopard frog, which has been found in certain populations from the north-central region of the United States, particularly Minnesota. This variant frog, known as bird's eye, is conspicuous by the absence of the customary leopard-like spots on the back. When first discovered in 1922 by Dr. Weed, then at the University of Minnesota, the non-spotted frog was designated as a new species, Rana bird's eye. However, the results of breeding tests in recent years have revealed that the bird's eye or non-spotted frog is a simple Mendelian dominant to the common spotted or wild-type pattern. Hence, the bird's eye pattern is due simply to a dominant mutant gene. One question that immediately comes to mind to the developmental geneticist is, how does the bird's eye gene express itself? How does it cause its effect? The bird's eye gene somehow suppresses the formation of the black dorsal spots on the back. Now, the spots of the wild-type frog are essentially compact clusters or aggregations of melanophores. One might assume that the non-spotted condition of the bird's eye frog is due to the absence of melanophores. This, however, is not the case. The next slide shows a whole mount preparation of a portion of the dorsal skin of the bird's eye frog. Melanophores are present in the bird's eye frog, but they are not clustered together to form spots. The failure of pigment cells in the bird's eye frog to aggregate into spots may be the result of some defect in the melanophore itself or in the tissue environment in which the melanophores differentiate and migrate. The question thus is whether the bird's eye gene alters principally the intrinsic properties of the melanophore or acts indirectly by modifying the tissue environment in which the pigment cells migrate. As seen in the next slide, the potentialities of pigment cells can be tested by the transplantation of neural fold tissue. It is well known since Duchenne's decisive experimental studies 3 decades ago that the pigment cells of amphibians originate from the neural crest of the embryo, which are clusters of cells that proliferate from the inner surfaces of the neural fold. An ideal implantation site, as shown here, is the mid-ventral surface of the embryo, a region which permits an investigator to follow the fate of the neural fold graft with relative ease. Accordingly, one lateral neural fold from the right side of a wild-type embryo was extirpated, and the excised piece was inserted through a slit in the ventral surface of a bird's eye embryo. The reciprocal experiment was also performed in which a bird's eye neural fold was transplanted into a wild-type embryo. By this means, one can ascertain whether the grafted pigment cells would develop autonomously or be modified by the environment of a different genotype. As seen in the next slide, in the early post-operative stage, the relatively bulky transplant is evident externally as a moderate bulge in the abdominal region. As the embryo grows into a feeding larva, the mid-ventral bulge flattens, and, as seen in the next slide, the graft, whether it be a wild-type genotype or a bird's eye genotype, self-differentiates and becomes the center of a circular patch of melanophores. This melanophore-laden patch stands in sharp contrast to the silvery adjacent regions of the host, colonized by white iridescent guanophores. The circular graft area has all the aspects of a transparent piece of dorsal skin. In other words, pigment cells differentiate in the graft as if they had not been transplanted. Of course, as seen in the next slide, cellular types other than pigment cells arise in the graft. The entire graft area is shown in A. Selected portions are enlarged in B and C. The embryonic implant contains presumptive mesoderm, which self-differentiates into skeletal muscle and cartilage. Nervous tissue, particularly ganglion cells, which owe their origin to the neural crest, are evident in B. Needless to say, the skin and internal regions are heavily populated with melanophores, as seen in C. The graft is normal and healthy in every respect. The surrounding host tissues do not adversely affect the processes of self-differentiation within the graft. This is the typical picture during early larval development. During mid-larval development, an unexpected phenomenon occurred. Surprisingly, the differentiated graft tissue degenerated. Blood vessels within the graft became dilated and engorged with erythrocytes. The skin of the graft area appeared swollen or puffy, and inflammation associated with the rupture of capillaries. Vasodilation was invariably followed or accompanied by disintegration of graft pigment cells. The sequence of external degenerative changes in pigmentation in the graft is shown in the next slide. Part A of this slide shows the circular patch of graft chromatophores in an initial stage of disintegration. As graft chromatophores disintegrate, host guanophores invade the graft area. As shown in B, the graft region loses its once circular appearance as host guanophores migrate into regions formerly occupied by graft chromatophores. Part C shows the small remains of the graft in a near final stage of deterioration. Reconstitution by host pigment cells is generally complete before the close of metamorphosis. In the newly transformed frog, the ventral surface is uniformly covered with a thick mat of guanophores without any evidence that a graft had previously been present. Histologically, pathological changes are also prominent, as seen in the next slide. In the preparation at your left, labeled A, may be seen the formerly normal piece of graft cartilage in an incipient stage of degeneration, slowly being totally digested. A conspicuous feature of the breakdown of the cartilage is a thick accumulation of lymphocytes around and heavily infiltrating the cartilage. The graft skin shown in B may be compared with a normal piece of ventral skin as seen in C. In the normal skin, represented by C, the deep layer of the dermis is composed of loose fibroelastic connective tissue. This contrasts with the bulky mass of connective tissue in the graft dermis containing numerous lymphocytes. Eventually, all cell derivatives of the embryonic neural fold graft degenerate. These events have all the earmarks of an immunological response on the part of the host. One would, however, be initially hesitant to suspect a host immune reaction, particularly since an extensive literature informs us that an embryonic graft should persist as a consequence of its early introduction and long residence in the host prior to the maturation of the host immunological machinery. Yet it can be demonstrated that the embryonic neural fold allograft does confer immunity and not tolerance. The evidence bearing on this point stems from the fate of subsequent skin grafts from the same donor to the same host in later life. The experimental procedure is shown in the next slide. In amphibians, even though the donor embryo had been deprived of a lateral neural fold, the donor embryo is able to regulate very nicely and transform into a juvenile frog. Hence, when both donor and host embryos had metamorphosed into young frogs, the host frog received the skin graft from the same donor that had earlier contributed the neural fold graft. The results may be tersely stated. These skin allografts in the host frogs were rejected with typical second set reactions, rapidly and vigorously. This statement is documented in the next slide. The median survival time of first set skin allografts at 20 degrees centigrade in control frogs, the first line, is 18 days. Second set skin allografts in the same control frogs are destroyed as expected within a relatively short period of time, specifically 11 days. In the experimental frogs, the last line, which had previously been exposed to the neural fold grafts, the skin allografts exhibited typical second set reactions. The median survival time is 12 days. The accelerated rejection of the test skin allografts indicates that the host frogs had been sensitized by the previous exposure to the embryonic grafts. When these results were first reported at a meeting, several embryologists in attendance were somewhat dismayed by the findings. Many investigators, particularly those working with salamander embryos, had observed that embryonic primordia may be successfully allotransplanted without any subsequent antagonism between host and graft tissues. Many of my colleagues suggested that the disintegration of the graft might have been due, at least in part, to the unsuitable nature of the tissue terrain of the ventral surface of the embryo. In other words, the ventral terrain of the embryo provides a detrimental environment for the maturation or maintenance of cells that arise from the dorsal neural fold implant. To discount this possibility, I enlisted the cooperation of my colleague at Tulane University, Dr. Robert McKinnell. May I have the next slide, please? McKinnell had studied with Robert Briggs and Tom King, whom, as you know, pioneered the now famous technique of successfully transferring a diploid nucleus from an embryo into a previously enucleated unfertilized egg. The diploid nucleus from a blastula is capable of promoting normal development of the egg. The technique of nuclear transfer affords a unique means of obtaining genetically identical embryos. An isogenic group is established when diploid nuclei from a single donor blastula are each transferred individually into several enucleated eggs. Accordingly, McKinnell and I collaborate on the type of study that is portrayed in the next figure. Several genetically identical embryos were produced by the technique of nuclear transfer. For ease of presentation, one pair of identical embryos is shown in the illustration. When this pair reached the neural stage of development, one lateral neural fold of one embryo, as in the previous study, was transplanted to the abdominal region of the other embryo. This was followed, when both embryos metamorphosed into young frogs, by the reciprocal transplantation of dorsal skin. Both the embryonic and dorsal skin allografts, rather isografts, should thrive and persist if the pair of frogs are indeed isogenic. As seen in the next slide, the neural fold isograph did flourish and persist through and beyond metamorphosis, and in fact indefinitely. The individual shown here is but one representative example. The pigment cells within the graft faithfully reproduced a miniature dorsal spotting pattern on the abdominal surface. Moreover, as seen in the next slide, grafts exchanged between members of an isogenic group were perfectly viable. As revealed by the enlarged views, the square pieces of dorsal skins blended in so well with the host skin that the borders of the skin grafts are scarcely perceptible. Notwithstanding these findings, some embryologists persisted in the view that the success or failure of the neural fold allotransplant might have still been conditioned by the site of transplantation. In other words, as some of my colleagues then contended, the results might be different if the embryonic neural fold material is transplanted orthotopically, that is, to its anatomically natural position in the host embryo. Accordingly, as seen in the next slide, one lateral neural fold from the right side of one embryo is extirpated and replaced with a corresponding fold from another embryo. When the paired neural folds in the host embryo united along the dorsal midline, the resulting neural crest was of dual character. The right side was of donor or graft origin, and the left side was of host or normal origin. This type of embryo is appropriately called a chimeric crest embryo. As seen in the next slide, when the host embryo metamorphosed into a juvenile frog, the greater area of the skin of the right hind limb was actually devoid of pigment cells. The ghostly appearance of the right limb is a telltale sign of degeneration of graft pigment cells. With the disintegration of graft cells, the host is able to repair or reconstitute a pigmentary pattern in the back region. However, the host pigment cells are unable to migrate into the right limb, and a large area of the limb remains permanently pigment-free. Once again, it can be demonstrated that the disintegration of the neural fold allotransplant is the outcome of an immune response elicited from the host. Subsequent skin allografts from the same donor are fully rejected in an accelerated fashion. With these results, we became even more curious as to why embryologists in the past had failed to detect transplantation incompatibility. Conceivably, success in the past might have stemmed from the amount of embryonic material transplanted. In the next experimental series, the amount of neural fold material allografted was increased twofold. Specifically, as illustrated in the next slide, both lateral neural folds were removed from one embryo and replaced with the corresponding right and left folds of another embryo. As shown, the neural folds were exchanged reciprocally between the two embryos. Such experimental embryos are termed foreign crested, as the neural crest material in the trunk region is completely alien to the embryo. As seen in the next slide, the pigment cells of graft origin do survive through the larval period, and at metamorphosis give rise to a pigmentary pattern characteristic of its genotype. The anterior portion of the body displays the non-spotted or bird's eye pattern of the host, and the trunk region exhibits the wild-type pattern of graft origin. The conspicuous pigmentary pattern of the posterior part of the body is sufficient documentation in itself of the viability of graft tissue. To complete the proof, a piece of wild-type skin from the experimental partner, that is the other member of the pair that had originally donated the wild-type neural crest graft, was transplanted onto the host or anterior portion of the frog. As is evident from the enlarged view of the patch of skin, this second or test allograft from the same donor is perfectly viable. Thus, the host's reactive system can only be suppressed by a large dose of transplanted embryonic material. The threshold level for invoking tolerance is at or very near the amount of graft tissue contained in 2 complete lateral neural folds. Tolerance cannot be induced when the population of graft cells falls below the critical concentration embodied in both lateral folds. If one fails to incorporate the complete tissue of the 2 lateral neural folds in the host, as is often done by my graduate students, then one witnesses a dramatic example of rejection as shown in the next slide. The subthreshold amount of graft tissue had disintegrated, and host wild-type cells were unable to reconstitute a wild-type pigmentary pattern in the posterior part of the body. In essence, the major finding that emerges from these series of experiments is that an inurine embryo can be rendered tolerant to the sensitizing properties of an embryonic allograft by increasing the quantity of transplanted tissue. It is not true, at least in the leopard frog, that an embryonic transplant is not subject to an allograft immune reaction simply by virtue of its residence in the host long before the host had acquired the faculty of immunological response. An embryonic allograft can induce either immunity or tolerance, the given response being largely dependent upon the amount of antigenic stimulus. A small allograft is almost invariably rejected. A large allograft comprising 2 complete lateral neural folds survives and persists indefinitely. It is important, I believe, to recognize that the difference between a small and large allograft is in amount only and not in kinds of cells. The same kinds of cells survive and persist only if they are present in large quantities. In other words, the same kinds of cells can either stimulate or suppress the host's immunologically competent cells. This suggests that the state of tolerance is not attributable to the property of the host immune system to recognize the graft's antigenic determinants as part of its own system. Rather, in tolerant animals, it would appear that the graft cells continually remain foreign to the host. How these alien cells, however, inhibit or block the host reactive system is still an open question. Although the donor and host cells are mutually antigenic, they are mutually acceptable. It can now be shown that the state of tolerance to massive embryonic allografts is accompanied by blood cell chimerism. The demonstration of blood cell chimerism was achieved by bringing into play two experimental procedures that are uniquely suited to the leopard frog, namely the induction of triploidy and the in vitro cultivation of peripheral leukocytes. Thus, both lateral neural folds of a triploid embryo were transplanted orthotopically onto a diploid embryo. If mesodermal elements of the triploid neural fold graft differentiate into blood stem cells which seed the hemopoietic sites of the host, then an analysis of the chromosomes of cultured leukocytes of the host should reveal the presence of both diploid and triploid embryos. Very rapidly then, the next slide shows the manner in which triploid embryos were produced. Normally, as shown in the top row, the second meiotic division is completed upon entry of the sperm with the consequent elimination of the second polar body. When, however, the egg shortly after insemination is subjected to hydrostatic pressure, the formation of the second polar body is suppressed and the resulting egg is triploid. As seen in the next slide, the eggs were pressurized in a Carver hydraulic compression press. A high incidence of triploid resulted when the eggs 5 minutes after insemination were subjected to a pressure of 5,000 pounds per square inch for 6 minutes. When the diploid host larva bearing the tolerated triploid neural fold allograft transformed into a juvenile frog, drops of whole blood were taken from the femoral artery of the thigh and the leukocytes were cultured. The composition of the culture medium is shown in the next slide, which is simply a modification of the culture medium that is used to karyotype or to grow human leukocytes. As seen in the next slide, the chromosome preparations of the cultured leukocytes of the tolerant host frogs contain both diploid and triploid metaphase plates. This figure shows a microscope field with 3 metaphases, 2 of which are diploid, containing 26 chromosomes, and the other triploid containing 39 chromosomes. The foreign or donor-type leukocytes may constitute as high as 8% of the blood cell population of the host. The chromosome complements are clearly diploid and triploid, as revealed by karyotype analysis shown in the next figure. A tolerant host frog is thus a blood cell chimera, in that it produces 2 kinds of blood cells, its own kind and that of donor origin. It would seem then that the donor-type blood cells which colonize the host act to promote, if not initiate, and maintain the state of tolerance. Tolerance accompanied by blood cell chimerism was also evidenced when portions of embryonic tail tissue, rather than neural fold material, were reciprocally transplanted between unrelated embryos. The tail grafting experiments were done by Dr. Brian Gebhardt, a colleague of mine now at the University of Florida Medical Center. Here again, the rejection or persistence of the tail allograft is governed principally by the size of the transplant. The next slide shows an embryo with a small tail graft. The small transplant comprises only the posterior 1/4 of the tail. As seen in the next slide, the small tail allograft has been reacted upon adversely by the host. The tail graft is no longer present, leaving the host with a slightly shortened, blunt tail. When, however, seen in the next slide, the host embryo receives an amazing transplant of a whole tail from the cloacal pore to the tip, the large transplant induces a state of tolerance. As shown in the next slide, the large tail transplant persists and becomes very much part of the host larva. Now, it might have been expected that when the tolerance-inducing large tail graft was broken down and resorbed by the host at metamorphosis, the host tolerance to test skin grafts from the original donor would be lost. However, this is not the case. Donor origin skin grafts were tolerated indefinitely by the host in post-metamorphic life, even though the initial large tail graft was no longer present. May I have the lights, please? Lastly, the outcome of the various experiments leads me to enunciate one important point. There is no evidence in the leopard frog, there's no evidence that the leopard frog, in its development, passes through a transitional stage from tolerance responsiveness into immune responsiveness. The data presented here cast doubt on the notion of a null period. None of the host frogs behaved toward a test skin graft as if they had not received the prior treatment of engrafted neural fold tissue. Host frogs, which in very early life, as early as 48 hours after fertilization, received the neural fold allograft, invariably in later life either became sensitized or rendered tolerant. Thus, in the leopard frog, there is no period of life in which the effect of a prior treatment is such that the adult behaves as if it had received no prior treatment at all. In other words, an embryonic allograft has important, perceptible consequences for a subsequent transplant from the same donor. Thank you.
Facts reported by Volpe is the lack of this null period from the immunological point of view during the grafts from the embryo to the adult of Rana pipiens. Histocompatibility antigens are either immunogenic or tolerogenic, even if the transplantations are performed so early as 48 hours, I think, after fertilization. This could suggest that in amphibia, the histocompatibility antigens differentiate very early. And in fact, that is also shown by the reverse experiment which has been performed by Simnet, who has observed a second set allograft rejection when grafting embryo in adult of Xenopus laevis presensitized by an adult skinned allograft. Then, as soon as the animal has reached immunological competence, antigens behave like injected to fully competent adult. We would like to know when the first interaction between antigens and competent cells occurs. Are there such cells as soon as the earliest transplantations are performed, or do the tissues on the antigens wait and how for the first immunologically competent cells? I feel that some light could be thrown on this problem by looking at the first cells which interact with the antigens during the antogeny of the animal. I would like to report on the larvae of a toad, Alites obstetricans, stimulated at 25 degrees centigrade with not with histocompatibility antigens of the species, but with mammalian erythrocytes, which allows some studies at the cellular level. May I have, no, thank you. Just in a moment. May I have the light, please? We have been using for that the techniques of immunocytoadjuvants, which permits detection of antibody-producing and also antibody-carrying cells which form rosettes which sheep or human erythrocytes use as antigens. Rosettes-forming cells can be counted in suspension of lymphoid cells. We also use local hemolysis technique in liquid medium, which permits the counting of hemolytic antibody plaque-forming cells. And we have followed by this means at the cellular level the ontogeny of the antibody-producing capacity in the spleen of tadpoles immunized with sheep or human erythrocytes. Assays were performed during the 2 first months of life at 25 degrees centigrade, when the number of cells increases roughly from 10 to the 2 to 10 to the 5. These experiments have shown the main following fact. I would like to
I hold that one for you?
No, no, thank you very much. Well, before any immunization, all the tadpoles have shown a background level of natural rosette-forming cells, but no natural plaque-forming cells. RFC are rosette-forming cells, and in non-stimulated animal, no background plaque-forming cells. That's point one in non-stimulated animal. Then, when I stimulate animal, and I may say that whatever the size of the animal, there are always a background level of rosette-forming cells, and whatever the number of cells in the spleen. Now, after immunization. Two case events. First case, when only the animals having roughly 6×10⁻³ spleen cells are able to elicit an immune response which is detected by the appearance of PFC and the increase of the number of rosette-forming cells. While on the other one, the animal having 5×10⁻² to 6×10⁻³ always show only a background level of natural rosette-forming cells. While the number of spleens increases from 5×10⁻² to 6×10⁻³, there is a period then where no immune response, but only the background level of rosette-forming cells can be detected. Meanwhile, allografts of the tail were not rejected, and I think that this period could correspond to that one mentioned by Volpi, during which the grafts can self-differentiate. And we have carried on studies to look to the nature of those rosette-forming cells. These cells look like lymphocytes and do not behave like macrophages. During the growth, the number of rosette-forming cells with human erythrocytes increases from 4 to 5 in spleens of 500 cells to about 100 in spleens of 10⁵. And under such conditions, the ratio, the number of natural rosette-forming cells per the number of total spleen cells decreases roughly 10 times from about 8 per 1,000 to 0.8 per 1,000. Recently, we have found that a percentage of the natural rosette-forming cell was thymus-dependent, as it is shown on the table. Please, can I have the slide? Here you have the age at which the thymectomy was performed. Here the number of cells in the animals, and here the normal background level of natural RFC, of natural rosette-forming cell with human erythrocytes. Here, after thymectomy, you can see that there is a very great decrease, especially in the very small animal, and here we have the average inhibition due to the thymectomy. May I have the light, please? Thank you. Thus, immune response were also impaired. Moreover, to a certain extent, The intensity of the immune response for each kind of erythrocyte has been looked to be linked to the background level of rosette-forming cell for each kind of erythrocytes. This has been published recently. It will appear, I hope, very soon. For those reasons, we think that a certain part of the natural rosette-forming cells are involved in true immunological processes and possess some degree of immunological competence. It's likely that histocompatibility antigens, if present, can also combine with such cells, since we have shown that some cells of a graft can reach the spleen and the liver of the host during the first week after transplantation. And the liver do contains natural rosette-forming cells. Well, despite the fact that erythrocyte antigens and histocompatibility antigens are not exactly homologous, I think that both experiments of Volpi and ours complement each other. We have an evidence that during a very early and supposed formerly neutral period, there are cells able to react with antigen and which must be involved in the immunological stimulation. Now, the problem which arises are the following. What is the origin of these natural rosette-forming cells, and is the binding between the antigen and the natural rosette-forming cell specific? Well, before new experiments are done, we can speculate only on the nature and to explain the nature of these natural rosette-forming cells. They can be explained in several manners. First, they could be natural antibody-producing cells. They could contain, without excreting them, antibodies against microorganisms having cross-reactive antigens with erythrocytes. These cells would appear following a random stimulation after hatching in the non-germ-free water where the tadpoles live. Secondly, they could be also considered as antigen-sensitive cells present in the spleen or other lymphomyeloid tissues. The first point seems to us difficult to accept. If natural rosette-forming cells are due to a previous unknown cross-immunization, it would mean that animals are already immunologically active, and we should expect an anamnestic response with erythrocytes when using stimulation later stimulation. Yet we could not detect any acquired response at this time, only the background, never plaque-forming cells. The second point seems to us more attractive, and that's the kind of cell forming natural rosettes should represent a primitive category of cells, since they exist before the ability to produce detectable antibodies arises. They could come into contact with antigens very early, when they are still in a very small number. For instance, in Volpi's experiments, they could be in contact with the transplanted tissues as soon as the first cells appear in the spleen or even before. Under these conditions, we may wonder whether the bindings between the cells and the antigens are specific for the antigens. The number of lymphoid cells is very small in amphibian embryos and larvae, much less than in very early larvae, less than 500,000. If the binding of some antigens is specific, and if one cell is precommitted to react with only one antigen, as postulated in the clonal selection theory, young tadpoles will have a set of antigen-sensitive cells which will interact with a very few antigenic determinants like those of erythrocyte antigens, and probably less than 10 to the 3, very few, because of the high percentage of natural rosette-forming cells. Well, under such conditions, we do not understand how other antigens would be processed. If the binding of the antigen is not specific, or if it just reveals the mechanisms of self-recognition, it leads, however, to further responses of apparently specific acquired immunity. For instance, in the case of allografts, this has been very well shown by Hildman and Haas in 1959, or immunizations with various antigen. The specificity has been looked at by Evans in the case of T2 phage and bovine serum albumin, and by ourselves with erythrocytes. This specificity is known to occur now in several cold-blooded animals. Then, the hypothesis of a mechanism involving an increase of the number of antibody-producing cells due to divisions of pre-committed precursors seems not to fit very well with the results obtained in the Alites obstetricans larvae. Thank you very much.
These two papers are now open for discussion. Entertain questions at this time. Okay, Dr. Ceppolini.
Not initially. I really was lucky to come here in time following the beautiful paper of Dr. Volpe. And it seems to me that beside the important problem of the null period, there is another, I think, important interpretation of his findings, that probably for having a tolerance, and we must put in quote tolerance, is necessary to have a continuous administration of the antigen. And possibly the interpretation partly misleading of the tolerance period is due to, let us say, non-fundamental aspects of the embryo, where, for instance, the provisional take is possible, and therefore, because there's a provisional take, and this take is able to administer continuously a small amount of antigen, therefore there is a state of, labile equilibrium between tolerance and immunity. There are cases also from man of this very labile status. For instance, there is a typical example of a twin chimera of the Ryovan type in a woman, where the presence of the foreign cells, blood cells, were rejected during pregnancy, and afterward was quite shown that woman had been tolerant, evidently, to the twin tissue for, I imagine, at least 20 years. I don't know at what age she was pregnant. Afterward, she was quite clearly shown to respond with the second set, which again is in line with Dr. Volpe findings.
Yes, Dr. Volpe.
I would only like to make one brief comment, and that is that I agree that in order for tolerance to persist, the tolerance-inducing antigen must continue to be present in the organism. I'm also suggesting that, as in the case of twin cattle and as in parabiotic frogs, that the tolerance achieved by implanting a massive embryonic allograft is associated with blood cell chimerism. I'm not suggesting a cause and effect relationship here. I'm simply stating that the two seem to go hand in hand.
Yes, Dr. Auerbach.
Dr. Volpe, is there blood cell chimerism in the single dose vesicle?
The amount of blood cell chimerism is that we detect in tolerant frogs. Is it on? Yeah. It's not on. In tolerant frogs, the amount of blood cell chimerism that we encounter ranges from about 2% to 8%. In frogs which reject a single lateral neural fold, we are not able to detect blood cell chimerism, but then again, one is always faced with the question as to whether there is a small amount that we are not able to uncover. But I would like to make the statement that there is no blood cell chimerism in animals that reject a single lateral neural fold.
Yes. Well, let me just carry this a bit further. Have you looked for chimerism prior to the time when you would have expected rejection? Certainly once you get rejection, you would have destroyed the chimeras if they exist. But have you ever looked, say, prior to metamorphosis to see whether at that time there's chimerism? Logically there should be half as much.
We discussed this last night. No, I have not looked for it prior to the time that the graft is rejected. And this is a natural follow-up of what we discussed. You're absolutely correct. We should look at for blood cell chimerism at the time the graft still persists. Except that that graft on occasion will be rejected as early as stage 3 or 4. So the graft rejection of a single lateral fold will vary in terms of the stage of development depending upon several factors, one of which is the degree of antigenic disparity between host and donor. If we can get one that is rejected relatively late, then we can culture the blood of an early larva and see whether they are chimeric.
I'm working. Okay.
We have time for about 2 or 3 more questions.
Okay, I wanted to make a comment on the dosage dependence of tolerance in the tall Bufo-Marinus. Thanks, Bill. One of my students, Ron Germain, and I have been using a Mitchison-type design of soluble bovine serum albumin given to adult Bufo-Marinus. And what we've found so far is that you can get high zone tolerance or paralysis. And the dosage at which this occurs, if you normalize milligram of antigen dosage per gram of body weight, comes out to be in the same region that Mitchison observed high zone tolerance in mice. And this brings up a question which I don't know how relevant, I would say how applicable it is. It would be nice if you could get low zone tolerance here. It would be nice if you can get it in the homograft situation. But it looks as if in order to get these homografts to take, let's say this prenatal tolerance, you need persistence of antigen, probably live dividing cells. So I'm going to ask again, sort of elaborate on Dr. Auerbach's question. Do you think it would be possible if you gave small enough dosages, this is Dr. Volpe again, of this foreign neural crest material, you might be able to get sufficient persistence, let's say it's ignored by surveillance mechanisms, that you can get a stable low zone tolerance?
We have not been able to achieve that. We have found that invariably, unless of course you transplant between siblings where there's a certain amount of antigen sharing, if there's very little antigen sharing, invariably a small amount of neural crest material will be rejected. Another experiment that we have in mind that is presently being pursued in the laboratory with the aid of Dr. Houghton is to transplant 2 neural folds or 2 complete neural folds into the ventral surface, which we have not done as yet. We anticipate that even if you transplant 2 complete lateral neural folds into the ventral surface, that may be rejected because the antigens are not being given a wide exposure to the host as when you transplant orthotopically.
Cohen, Rochester. I wonder if it's really necessary that you do use neural crest, a neural fold, as the tissue of choice. Might not it be better in terms of trying to quantitate numbers of blood cells involved to use presumptive blood islands from the ventral area?
It's not necessary to use a neural fold. It can be done with any embryonic piece of tissue. The only reason we use a neural fold because it serves as a convenient means of following the fate of the graft. But you can do it with non-neural fold material.
Can you do it with less amount of tissue with the blood islands?
With the blood islands. Good thought.
Really get the blood-forming cells
Right.
there.
Good thought.
Nick asked part of the question I was going to ask. What I did want to ask was really an embryological question. Namely, when you transfer these neural crests, you do it earlier than blood island formation. And therefore, I guess you're suggesting that neural crest cells, among the diversity of functions that they can serve in the embryo, can also produce blood cells. Is that right?
Yeah, as many of the old-time embryologists will tell you the neural fold material contains a considerable array of embryonic material, including mesodermal elements. And in the paper, although I didn't make it clear in my presentation, I suspect that the mesodermal elements of the neural fold implant are self-differentiating into blood cells rather than into somite material that they ordinarily would differentiate into.
Okay, we thank you for your discussion, and I think we must hurry on now to the next speaker, which is Dr. Myron Varsenko from the Department of Medical Microbiology and Immunology, University of California at Los Angeles.
Sit on now? Okay, thank you. Ladies and gentlemen, we've finally gotten to the last class of cold-blooded animals, and it's nice to have 2 whole days pretty much devoted to them. When I first got into this field, using the reptile, I thought that I would be able to perhaps help answer some of the questions that have been asked in mammalian experiments by using reptiles, which really are, in a general sense, the ancestors of both birds and mammals. However, I found I've been answering more questions in animals below reptiles than above them. And I really have no biases in either direction, so I think that's good. Very quickly, perhaps I can summarize some of the related areas of works on reptiles. In fact, I think I can do it in about 3 sentences. Those of you who aren't transplanted probably hate me, but I'd like to say that in general, reptiles produce antibodies to soluble and particulate antigens in very prolonged responses of generally very low titer. And these responses are temperature dependent. Okay, I just covered 10 years' work there. And there's more than one class of immunoglobulins in reptiles. Whether these immunoglobulins represent IgM or IgGs, as we have in the homeotherms or endotherms, is still not known. Another 10 years' work covered there. Okay, let's get into antigens, transplantation antigens, and transplantation. First of all, we know from work done way back at the turn of the century that turtles have blood groups. The painted turtle, for example, has 3 blood groups at least. Other work has shown that there are 10 species of erythrocytic antigens. Let's put it, that's a little bit wrong. There are 10 species in which erythrocytic antigens have been demonstrated. No serum protein alloantigens have ever been attempted to be defined. And finally, here we are at transplantation. Back in 1923, May discovered in what I consider a classic work that the false chameleon, Anolis carolinensis, rejects skin allografts in 60 to 90 days at 23.5 degrees centigrade. So here we have it, Nick, chronic rejection at a fairly moderate temperature back in 1923. Since that time, there have been several reports on rejection of skin grafts in reptiles, and the first slide sort of summarizes all that up. Leave the lights off, please. Okay, there are three orders of reptiles. These are the turtles. Here are the lizards and snakes, and here are the crocodilians. At temperatures around 25, and that between 25, say, and between 20 to 25, generally, we find chronic rejection throughout the spectrum, and we have variations, of course. Most of these reports, I might add, are either personal communications or very brief reports, and no one has ever extensively looked at this. My own work is with the snapping turtle that you see up here. First set allografts reject in 47 days with kind of a wide range, but this range does not overlap at all with second set rejections of 25. So I thought that I had a fairly good system here to attack some of the basic problems of temperature dependence and so on as related to the immune response, and more specifically the response against allografts. To answer Dr. Clem's question of yesterday, how do you graft a turtle, I have the following slides. Here you see a very sophisticated method of anesthesia called a restraining device. For those of you who are fanatic anti-vivisectionists, I'm sorry, but better them than me. There was a large area of skin on the snapping turtle, on the ventral surface, and as compared to a blanding turtle or a painted turtle, these two would be very hard to graft, but here you have skin exposed. So what I did was I made a graft bed on one side, took a piece of skin from the other side, put it there. That's the autograft control. This side was then ready to receive either an allograft or a xenograft from either of these two other species. And the next slide shows a typical rejection, actually my best rejection. Here you see a piece of skin grafted over the top of the, sutured over the top of the graft. Here's the same graft 17 days later. This is at 25 degrees centigrade. Here's the graft at 22 days after transplantation. Day 34, we have hemorrhaging beginning in the periphery of the graft. Day 44. Day 47, we have a scab. So, and that is what I consider my zero endpoint, the scab. And then the graft falls off and we have a healing over. I was a little bit amused by my mentor's comment of fantastic lymphoid cell invasion of his hagfish skin, and you'll see why in the next slide. All this black area are lymphoid cells, and this is biopsies of an allograft. At 12 days we have a little bit. This is the graft, this is the host skin. At 40 days, all this black area are just concentrations of lymphoid cells. And here's the host skin again, that's the graft. Large dermal thickening, which was observed in several other species. And this is what is a collagen pad. This is what we have left after the graft has been sloughed, and the mononuclear cells appear to remain and eat away at whatever is left of the collagen part of the graft. Okay, next slide. Here we have a xenograft that's being rejected. This is in about 25 days, I believe. We can see a hemorrhaging and inflammation around the periphery, and all this black area again is lymphoid cells. And one of the strange things here is that there appear to be no epidermal attraction between xenogeneic skin, no epidermal connection between the epidermis of the host and the epidermis of the graft. Here you can see the epidermis sort of beginning to go above the graft and then cutting down through here, and there are all kinds of variations here. So my criteria had to be adjusted to histological data, my criteria for the zero endpoint. Next slide. To summarize the first and second set data and throwing in data at 33, we find that first set allografts survive quite long. This is, I think, yeah, the mean survival time is 47 days, and this is the range from here to here. Second set allografts here, and first set allografts at 33, slightly longer than second sets at 25. And the same is true for xenografts. First set, second set, first set at 33. Okay, so we have a definite temperature dependence here. Now, I would like to, a little bit later on, go into the nature of this temperature dependence. And I think Dr. Auerbach, from our very short talk during one of the coffee breaks yesterday, has really put me on the right direction here. Next slide. Here we see at 10 degrees centigrade, an allograft after 100 days. Looks very healthy, no sign of incompatibility histologically. Okay, so we appear to have shut off something completely. Next slide. In looking at this phenomenon a little bit closer, temperature changes have strange effects on these grafts. Here's a 10-degree allograft for 100 days, no rejection. At 33, we get pretty fast rejection compared to 25. However, if you put the animal into a 10-degree tank after rejection has initiated, you essentially turn it off, and the graft survives at that particular stage. So here we have one phase of a different kind of inhibition. Here you appear to be turning off the cells that are already there. At 25 degrees centigrade, we get 47. If you keep an animal at 10 for either 50 or 75 days and then put it into 25 degrees centigrade, you get rejection in 36 days if you consider the day of transfer day 1. So the conclusion obviously would be that something has been happening during this period to make this one go in 36 days instead of 47 days once it's transferred. Well, there's a hang-up here, and that is mainly that there is the factor of acclimation. When an animal is put from 10 degrees centigrade into a higher temperature, its metabolic rate and perhaps immunological response tends to overshoot for several days before it comes back down to the 25-degree level. Okay, next slide. And here is a slide that sort of combines a lot of different things. And the main point here is that if you look at skin grafts, both allografts and xenografts, first set, at 4 different temperatures, 33, 25, 19, and 10, you find a big break in the slope of this curve at 25. Okay, the mean survival time is vastly different between 19 and 25. Okay, this is where the big change happens. And this is, as you will recall, pretty much what Dr. Hildeman showed in his goldfish study related to temperature. And Dr. Cohen and Dr. Kalman find similar breaks in their curves when studied in a similar fashion. Now, when you look at the latent phase, what I consider latent phase when there is no sign of lymphoid cell invasion, and compare it to the rejection phase where you begin to get lymphoid cell invasion, and compare the lengths of these phases, you find an interesting thing. The difference between 25, we'll just talk about allografts now. Forget this line. 25 degrees and 33 degrees. We find that the latent phase varies only by a few days. The main difference is in the rejection phase, the length of the rejection phase. Okay, so we've got a good 10 days difference in rejection phase and only 2 to 3 day difference in latent phase. When you get to 19 degrees centigrade, both phases are vastly affected. And at 10 degrees, we don't even get a rejection phase. So this break in the curve may mean that there is another factor coming into play. And I asked Dr. Auerbach what he thought about that, and like a good biologist, he said mitosis. And my immediate response was, why didn't I think of that? And that's what we may be having here. We may have, in the higher temperatures, inhibition of antibody production. And in the lower temperatures, we may have inhibition of mitosis, so that you never get the clone of cells that is competent to reject the graft. Yes? Did you? Oh, okay, no question. Okay, then let me make one other point here, and that is, it would be interesting to see if you can get a mammalian type of rejection by raising the temperature to 37. Now, animals don't survive well at that temperature. So what I did was I simply extrapolated the line, and assuming that it was linear between 25 and 37, and you can see that rejections would still be significantly longer than you would expect in mammals. This is just a hypothetical thing, because we don't have the data actually here. But you can assume that since the lines are breaking in that direction, that this line would probably tend to break further in that direction, and that there must be some kind of a minimal latent phase that is needed to induce immunity. And this is also supported by the fact that these two lines tend to come together at high temperatures, so that there must be a minimum latent phase. Okay, I did all that without my notes. Now, there was a lot of related studies to this. One of the studies that I find very intriguing comes from Dr. Auerbach's lab. Dr. Sidke and Dr. Auerbach found that spleen explants from adult snapping turtles are able to induce splenomegaly and hepatomegaly in vitro in explants from young snapping turtles, and that such explants from young snapping turtles are not able to respond against mouse red blood cells, but adult explants are able to. And they also found that there was quite a late maturation of immunity, of competence in the young snapping turtle. And since this was the animal that I was using, I thought I would, actually this data was coming in already. We had separate ideas here, but they turned out to confirm each other. And the next slide shows my study on the maturation to reject skin allografts and xenografts. Here we have newborn animals rejecting between 90 and 100 days. And by the time they're 1 month old, this is significantly decreased mean survival time. And between the ages of 4 to 6 months, we have exactly the same mean survival times as we have in adult. Now, these animals got skin grafts from animals of the same age, so that the control here had to be testing the antigenicity of a newborn skin. And we found that transplants grafted from a newborn to an adult rejected exactly as fast as adult to adult. So that we do have a close correlation here. Studies from Wisconsin also showed, well, that's what the splenomegaly and hepatomegaly really show, that you could induce a graft versus host reaction in vitro. And it would be interesting to do it in vivo. We have not done that yet. And one of the haziest areas in the phylogeny involving reptiles is the occurrence and morphology of lymphoid organs in reptilia. A number of people have found bursa-like organs in the snapping turtle and other reptiles. Or maybe it's just the snapping turtle. I don't know. Tonsil-like organs were found in the alligator. Other reports have said that there are really a lack of lymphoid organs in the reptiles. So we, Dr. Cooper and I, looked at some lymphoid organs in the snapping turtle and found the following. Next slide. Not only do they have a thymus, a spleen, bone marrow, they also have confirming bursal equivalents. Perhaps these are what can be considered bursal equivalents. They have intestinal aggregates similar to Peyer's patches, lymph node-like aggregates in the axillary and inguinal regions, and there is something that looks like a tonsil around the trachea. The sphenodon was reported to only have one recognizable lymphoid organ, which is the spleen, and this came from Dr. Marcellonis' paper. What is generally, may I have the lights, please? What is generally lacking, I think, in the work so far in reptiles is to get more extensive data on lymphoid organs, evolution of lymphoid organs, evolution of immunoglobulins, transplantation antigens, and so on, and then to perhaps try to correlate these different areas to make some sense, some kind of a nice story that would involve all of these different factors, not only transplantation immunity, but immunoglobulins, lymphoid organs, evolution of antigens, and so on. And we have been doing a little bit of thinking in that direction. Thank you.
Thank you very much, Dr. Borysenko, for your nice presentation. I think you've been stimulated by this meeting, your discussions with Dr. Auerbach. I'm going to call Dr. Auerbach right now, and perhaps he'll elaborate a bit more on some of the things you mentioned.
Thank you. First of all, let me congratulate you on your work with the turtle. I admire it, and I've already told you this. I was very embarrassed some 3 months ago by getting an advanced manuscript. This just never happened to me before, to get a manuscript in time to think about it. And so when I talked to Dr. Borysenko, I said, well, you know, I've thought about it so much, I'm not even going to discuss your paper. And that wasn't, I didn't mean to be at all in any way negative about this, and now you've brought up all sorts of new things in addition. So I'm going to spend a little bit of time saying a few things about the work that we have done, only as a matter of perspective, discuss the reptilian work, again, only a little bit, and try to make some kind of a general comment, in which I guess the starting point is your paper. First of all, as you've mentioned, we have been able to produce antibodies to sheep red cells in vitro using turtle tissues. We have been able to get a graft versus host reaction. I must explain this to you, and I guess it reflects on my admiration for your work and reflects on Dr. Clem's question about turtle grafting. One of the reasons we chose tissue culture was very simple. We were going to inject some sheep red cells into the turtle, snapping turtle, and all I can tell you is that the snapping turtle has a number of problems. At least we have a number of problems with snapping turtles. Everything from being afraid we're going to get snapped to finding the fact we can't get the tail out, and if we do get the tail out, we can't find any blood vessels and whatnot. And so as a mouse tissue culturist, it seemed logical that there are two ways. One is to become a good reptilian biologist, and the other one is to kill the animal, get out the spleens, and culture them. And we chose the latter method largely by default. Although even killing turtles or anesthetizing them turned out to be a major problem, like overnight etherization and other things of this sort. Yeah. We have actually gotten away from turtles, as I think to some extent you have, and more recently have been able to use tissue culture methods with xenopus. And this reflects back on the earlier discussion of amphibians. And it turns out that in xenopus, too, with spleen tissue, it is possible to get antibody from, this has been done in collaboration with Dr. Rubin, It is possible to get antibody formation in vitro to sheep red cells, to mouse red cells. It is possible to get long-term antibody production. These are now primary immune responses. It is possible to immunize sequentially to two antigens, something which has not been possible in the mammalian system. We can culture xenopus tissue in the presence of one antigen, let this go for a while, add a second antigen, let this go for a while, and analyze sequentially what happens to an explant given these antigens, specifically mouse red cells, sheep red cells. We're now turning to other antigens. I have no reason to suspect that the graft versus host reaction, which works well in the mouse and chicken and in turtle, should not also work in amphibian tissues. I think the in vitro systems offer some opportunities here to get away from some of the problems that we're facing with lower vertebrates. So much for data, and I'd like to turn to the idea that you've started with on this temperature sensitivity in studies and expand on it just a bit, as an excuse for pointing to some cautions. I think we have throughout this symposium been making some assumptions which are perhaps not quite valid. For example, if we talk about temperature reactions, as you've already pointed out, we are talking about an extremely complex parameter. We're talking about optimum for enzymes, we're talking about optimum for cell division, we're talking about depletion of cells. And we put all of this together, and it's not at all surprising, I think it's encouraging as far as the animal is concerned, that we don't find a smooth curve, or that you have been able to demonstrate a break in the curve. I think if we knew more about it, there would probably be several breaks in the curve. And in fact, what we need is a lot more information. Temperature may be an extremely good way of finding places of interest in which to analyze immune reactions. Now, I think that what we've done here in the last day and a half, so far at least, is largely pointed out that a number of systems have specificity reactions, transplantation reactions, and immune reactions of various sorts. In the discussion of the preceding paper, I think you may have noticed, if you saw it almost slipped by in the speed of in the shortness of the time, that in the case of one amphibian, for example, an animal which doesn't really have enough cells yet to follow dogma of immunology is making antibodies, or what seems like antibodies. It's having specificity reactions. Now this raises, I think, the important point, and your paper does too, that I think we should look and ask ourselves questions. And then we should look at what is now a catalog of animals, of species, and decide how to answer the questions. And not worry terribly much about whether we have to work with a monkey embryo, as I've done it on occasion, or with an adult tilapia or bullhead, which we've also done on occasion, but rather ask which species, which animal, at what particular time can answer the questions. Now, I think there are some cautions. For example, we make the tacit assumption that we're going to have an evolutionary sequence. But I think your first course in evolutionary biology tells you that there are many different ways that animals have of solving their problems. There are such things as convergent and divergent evolutionary pathways. If it is true, as Lee Hood has suggested, that the immune system may involve 1 million genes for gamma globulin alone to explain antibody variability, and he justifies this by saying that 0.1% of the DNA is not too high a price to pay for survival, it may be that only 500,000 of these genes are involved in immunoglobulins, and the other 500,000 may use 500,000 other ways of solving a problem. So one has to keep that in mind. I think if one wants to ask particular questions of the amphibians or of the reptiles or the birds, or well, I can see a number of them. Now, specifically, we have been interested in the origin of antibody-forming cells. Now, Pete Volpe's neural crest experiments give us a possible clue. I think perhaps even more importantly, if you want to study the yolk sac origin of antibody-forming cells, you can't use amphibia in the same way. But we can talk about reptiles and say, well, if we want to do some experiments with embryos, the reptile is an ideal animal to work with for that question. I think it should be used. I think in another way, we want to talk about what happens when an animal has an early exposure to foreign things. In that case, we don't want to use an amniote. An amniote is protected from its environment. In that case, an amphibian is a beautiful thing to look at, and it may well be that what we've heard just a few minutes ago is an example of a very early necessary immune system, because the amphibian is not as well protected from the environment as, say, the reptilian embryo is. There's no reason the turtle has to make responses quite as quickly. It can survive. We know the turtle does not make antibodies in winter, at least not the Wisconsin turtle. It goes through I won't say hibernation, but a quasi-hibernation state. This is not just a matter of cold temperature, because the spleen taken from a November turtle won't make antibodies in vitro at 30 degrees. Now, it may not have enough cells. In the case of the hamster, which is cold adapted, does not make antibodies until we've given it a day to recover. It can recover in the presence of other tissues. It's missing a mitogen. It's missing some tissue contribution. It's inhibited some way. So again, I think perhaps the main point that I want to make is I think there are a tremendous number of questions in immunology that need answering. And much as I like to say the mouse can solve problems, and I've thought so for a long time, there are a number of questions which clearly cannot be answered with a mouse. There are some questions which can't be answered with amphibian. There are some questions that can't be answered in turtle. But I think it's time now to look at the whole spectrum and choose the right systems.
Thank you a lot, Dr. Auerbach. I'm certain there are many things that we can do with turtles. As a matter of fact, you can immunize them if you want to prepare them properly. And I recall an experiment of Dr. Ballmer's in which he was studying insulin shock in turtles. And he merely took a little square out of the breastplate directly over the heart, and then he made his injections of insulin directly into the heart. He had some turtles left over, and he said, "Would you care to immunize them, Stormont?" And I said, "Yeah." I happen to have some sheep red cells around. These weren't domestic sheep. They happened to be sheep family tar goats at the San Diego Zoo, and I did inject them, but I had to wait quite a while for antibodies. But I learned that this was temperature dependent. But I got some very excellent antibodies against sheep red cells, and some very highly specific antibodies, as proved by absorption experiments. So I often thought of going back to turtles to use in some of my immunization experiments, but we just didn't have facilities to handle them at Davis, California. But I may come back to those someday. Now, we're open for discussion. Dr. Cohen will take it first.
Dr. Auerbach, I'm very interested in your observation that the turtle, the hibernating turtle, doesn't make antibody. How do you think the animal copes then, all the cold-blooded vertebrates and the hibernating animals, with their environment? There are pathogens that live at these temperatures. Do we in fact find, as I've heard from some people, that during the months of hibernation of frogs, that there's a great deal of animal death? Does anybody here have any information on that?
Is Dr. Yaroslav here still? Why do you?
Well, particularly with the hibernators, it's a marvelous situation. They have a spontaneous recovery from many parasitic infections. So when they go into hibernation in the fall, they have a heavy intestinal parasite load. They wake up in the spring and they're fairly clean. And this is true with trypanosomes and with some bacterial infections. Now that I'm up here, I might as well mention something fascinating about what Dr. Borisenko said. He had a protocol there that was very much like one that I set up in with my hibernating squirrels. Can I go to the board? This protocol in which he alternated high temperature and low, we did the same. We were following antigen disappearance in the ground squirrel, and everything he found was the same thing we found. And there was one thing we did that he started to do and didn't do, but I suspect he probably has the information. Now, you know the normal antigen disappearance curve. There's this period of equilibration, metabolic disappearance, and then rapid disappearance as antibody forms. And so when we injected labeled BSA into the hibernating animal, we had no disappearance during hibernation, and upon arousal we had a disappearance that reflected a 7-day latent period, and then we had the rapid disappearance. Now, all this is in agreement with what you've said. Now, we did this experiment. We injected the BSA, and for 4 days we followed its disappearance, and it was normal. Of course, it was a normal ground squirrel. We put him in hibernation for 2 weeks, and there was no disappearance of antigen during this time. Now, when we take him out, you can expect either you'll have a 3-day latent period, 3 plus 4 being 7 to make it normal, or if something happened during this interval, that is further development, would be less than 3 days. Now, 6 animals in which that slept well, we had 2 that came out producing antibody, indicating that during this interval they had completed the response. But then we had 4 of those 6 that had a 7-day latent period. The normal latent period, it was as if these 4 days had never happened. And so now I ask you, when you did this experiment where you had them in at 33 degrees for, I forget how many days, but it was considerably less than rejection time, and kept them at 10 degrees for 100 days, you saw no rejection. But when what did you do? Did you bring them back to temperature, and did you notice how long it lasted?
We did not bring them back to the 33-degree temperature. We just biopsied the allograft after the observation period and compared it to the way it looked before we put it at 10 degrees centigrade, and found that it pretty much stayed at that level. There was no significant change and no disappearance of lymphoid cells, just no significant, it seems to have just stopped and stayed
Yes,
that way.
well, you'd kind of expect that in the cold.
But we did not change the temperature after that, so I don't know what would happen then.
Yeah, see, we've done, of course, a whole series of different experiments with the hibernators, and it would appear from our results that what happens is during long periods of hibernation, these cells presumably can't divide as far as we know, and my guess is that they may become memory cells as long as the antigen is present in large amounts, circulating antigen. Apparently, when they wake up, they do not immediately start a second-day response, even though you say, well, the antigen's there, why don't they? But apparently it's this continuous circulating antigen that blocks it. But then if they are awake long enough for the antigen to disappear and they can go through a whole cycle, they now have a very good secondary response, which would require that what had happened before would have formed memory cells. And your system is beautifully like ours, except you can keep them at 10 degrees. And with hibernating animals, the trouble is that they wake up spontaneously, and you shoot the whole thing once they do.
Okay, Dr. Cole.
I'd like to make one comment on temperature effects of transplantation immunity in salamanders and pose a question to the audience. We've kept animals at 15 degrees centigrade, and the response, in most animals, there's no response, as indicated by histological evaluation, and as indicated by a very sensitive assay system of individual pigment cell death. But in some animals, individual melanophores were seen to undergo destruction on allografts, but not on xenografts, on allografts, but not on autografts. So does temperature then totally suppress mitosis? Is the mitotic rate of a lymphocyte or an antigen-recognizing cell variable? In other words, how can we allow the escape of some cells, the obvious recognition of the antigenic stimulation, but the very, very slow infiltration of the graft, if we assume that mitosis is totally suppressed?
The fact that the graft heals in at 10 degrees centigrade must indicate that there is some metabolic activity, including mitosis, perhaps.
In other words, the mitotic rate is maybe altered.
Yeah, maybe altered to the point where
But it's not an all or none phenomenon.
It may depend on the animal, I think, also. But I just like to point out at this time, getting away from your question just for a second, is that Dr. Hildeman and I have found that hagfish, which are normally animals that stay at 10 degrees centigrade in their lifetime, do begin to reject allografts at 10 degrees centigrade, even though we have very, very prolonged survival. So perhaps someone else can answer that.
Dr. Auerbach, there's a young man that wants to ask a question behind you, so I'll let him ask his question first. Or maybe he wants to volunteer some information.
Nick, I think that what you have to consider is there are two slightly different aspects to this question. One is that even though an animal is poikilotherm, that doesn't mean that it is all the same. For instance, a paper has recently come out from Australia pointing out that we should look at animals like fishes and amphibians much differently from reptiles because they are behaviorally warm-blooded. Secondly, you have to consider each organism with regards to its temperature range because, for instance, our annualfish, Cynolebius, at 15 degrees, they grow 40 to 60% faster than those at 20. So obviously, mitosis is occurring. So I think this is a little bit of elucidation on your question.
Okay, thank you, and we'll have one more question or comment. Dr. Auerbach. And then we're going to have a coffee break.
Well, your comment, I think, is extremely well taken. I think there's an additional point, and that is that we've been able to find that in, say, hibernating animals, something is produced which will inhibit antibody formation, and it apparently will inhibit cell division of certain kinds of cells, and not all. You can call it a hormone, if you will. It's located in the brown fat, and it's extractable and can be, I think, purified. But clearly, when an animal turns off antibody formation, it's an extremely complicated thing. One needs to look at turtles. Is there something in the circulation of the turtle which inhibits? We need to look at recovery from the cold. In the case of the hibernating hamster tissue, at least it seems to be a thymus-dependent recovery. I don't know whether in the case of amphibians it's a thymus-dependent recovery or in the case of the turtle. But I think, again, a word of caution. I think there may be hormones involved, and these will differ in different animals at different temperatures.
All right, we're going to have coffee break now, and I'd like to hurry the coffee.
I think there are two main criteria which call for a distinction between so-called primary and secondary lymphoid tissue. The first criterion says that primary lymphoid tissue, such as the thymus in all vertebrates, the thymus and the bursa fabricius in birds, are the first organs to become lymphoid during ontogeny by the immigration of stem cells, presumably from the embryonic yolk sac. In contrast to these primary lymphoid tissue, the secondary lymphoid organs, such as, for example, lymph nodes, Peyer's patches, tonsils, appendix, become lymphoid subsequent to the primary lymphoid organs during ontogeny. And it is because of this sequence in the appearance of lymphoid differentiation in primary and secondary lymphoid organs that the removal of a primary lymphoid organ results during ontogeny or prior to lethal irradiation in adult animals, results in severe functional and structural deficiencies in the secondary lymphoid tissue. It is the purpose of this paper to try to relate the structural and functional degree of secondary lymphoid organization in species of different evolutionary status with their capacity for antibody production. And the work I'm going to talk about has resulted from collaborative efforts between Mrs. Norby, Dr. Marcellonis, Dr. Ely, and myself. Before we start our survey, I would like to introduce you to the techniques, or the main technique we have used to study the immune response at the cellular level from the morphological and from the quantitative point of view. Use was made of the phenomenon of bacterial immunocyte adherence. A cell which secretes antibody directed against a particular strain of bacteria will cause these bacteria to adhere to the cell surface. Such cells can be identified on cell smears, as in this situation. They can very readily be processed for autoradiographs. Here we have a labeled cell. This is a non-labeled antibody-forming cell, using tritiated thymidine as a label, as a precursor. Or these cells can be enumerated if present amongst other cells in the cell suspension. This is done by taking an aliquot, next slide please, and incorporating this aliquot of cells containing antibody-forming cells into bacterial nutrient agar. The whole is then plated on Petri dishes, and the Petri dishes are incubated at 37 degrees for a few hours. During this time, bacteria, coronae of bacteria around antibody-forming cells, will grow into bacterial colonies. These colonies can be stopped from further growth, they can be stained, and they can then be counted at low power magnification in a dissecting microscope under very carefully defined optical conditions. Let us now start our survey with the teleosts, with the representative of the teleosts, Persa fluviatilis, the perch. The teleosts belong to those vertebrates which have a minimal amount of clearly defined secondary lymphatic tissue in the presence of the splenic white pulp. If we immunized fish with 20 micrograms of the very powerful immunogen, polymerized flagellin from Salmonella adelaide, and we kept this fish at a temperature of 25 degrees, then we obtain immune response now measured at the cellular level, which starts, there are no antibody-forming cells at 1 day, but there are antibody-forming cells present at 2 days, and it was very, very interesting to see that there is virtually no exponential phase in this response. So we can't really speak of a peak reached by proliferative processes in this response. It seems as if these first antibody-forming cells appear as, in a very explosive way, they're just suddenly there. If we give a secondary injection here of the antigen, we obtain a secondary immune response, which we may call an amnestic response, but the fact that there were antibody-forming cells still present at that time, we can't really be sure that this has resulted from memory cells which have been fired off, or whether this has resulted from just further proliferation of cells, antibody-forming cells, which were there already. Now, because of this sudden appearance of antibody-forming cells, we had a look at the morphology of these cells. Next slide. And we found that they belonged to the category, and I have to say that between 80 and 100% of these cells, there was variation between between different animals, 80 to 100% of these cells belong to the category of small to medium-sized lymphocytes. That is as determined using criteria which are used in mammalian cytology. A small lymphocyte of a cell diameter of 8 or less than 8 microns, medium-sized lymphocyte 8 to 10 microns. And when we gave 3-hour pulses of tritiated thymidine, none of these small cells were ever labeled. And we concluded from this that
there is a high probability that these cells have in fact arisen by differentiation rather than by proliferation. Let us now go a step higher to the amphibians. Amphibians are vertebrates which for the first time in evolution show the presence of secondary lymphoid tissue besides the white pulp of the spleen, which have similarity with lymph nodes. These tissues are in the jugular region and are therefore called jugular bodies. They're very small. They consist of a reticular framework containing lymphoid cells and quite clearly definable macrophages. Let us have a look to the next slide, please. We have studied first the function of the secondary lymphoid organs, that is the splenic white pulp and the jugular bodies, in the toad Buffa marinus by injecting radio-labeled iodine 125 labeled polymerized flagellin into the toads. Here you can see the uptake of antigen on autoradiographic preparations of sections from the spleen around the white pulp in a region which, if it were a mammalian spleen, we would call the marginal zone. But if we now look at the jugular bodies, next slide, then we found a very, very high degree of antigen uptake. And the antigen was distributed throughout the tissue in a very uniform way. There was no suggestion that antigen distribution would have changed within the time of observation, that is, daily observations over a period of 5 days. Because we could not determine with what cells this antigen was associated in these jugular bodies, we carried out electron microscopic sections and processed them for autoradiographs. Next slide. And we did this with reference to antigen uptake in the cortical follicles of lymph nodes from the rat. And here you can see, if I wouldn't tell you which slide belongs to what species, I don't think you could tell. This is the toad, and you can see that antigen has been taken up in association with surfaces of cytoplasmic processes. And despite the fact that there were phagocytic cells present, we could only very rarely, and then we were never sure if it was really significant uptake of antigen. We could only rarely find antigen inside phagocytic cells. Here, as shown by Nossel, I think that's familiar to you, antigen is in the rat taken up in exactly the same way as in the jugular body of the toad. That is, antigen is attached to surfaces of cytoplasmic processes. Now, if this antigen taken up by the jugular bodies does induce immunity, then we would have to conclude that this immunity would have been induced by free antigen, and not that the immunogenicity of the antigen was mediated by phagocytic cells. We have no direct test on the toad, but I have an experiment I'm going to show you, which we have carried out in the mouse spleen, which showed us that to this antigen, phagocytic cells are not required. Next slide, please. We culture spleen cells from the mouse in vitro for 4 days in the presence of either sheep rat cells or our Salmonella adelaide polymerized flagellin. Here we have the original spleen suspension. This is the immune response to sheep rat cells. And in the very same culture, we have the Salmonella adelaide antibody-forming cells with this response. We have then removed from this original spleen suspension the phagocytic elements using the short modification of the Rabinowitz column. And doing this, we lose the immune capacity of the spleen cells, now free of phagocytic cells completely, with respect to sheep rat cells. But there is still immune response present to Salmonella adelaide antigens. Here we have the fraction which we have removed. This is the adherent fraction, that is, cells
adhering to the glass beads of the column. They are taken and tested for their capacity to respond. You can see that now they will respond to sheep red cells somewhat and to Salmonella adelaide. This indicates that we have also removed lymphoid elements together with the phagocytic cells. That's why we have a slight drop here. But I think we can conclude from these data that for sheep red cells we cannot say that phagocytic cells are not required, but we can definitely say they are not required for Salmonella adelaide antigens. Next slide, please, shows us what happens if we try to reconstruct the normal situation with respect to sheep erythrocyte immune competence. Here we have the control groups. The original taking 12 million cells, that's the response to sheep erythrocytes. Now we take the lymphocytes, that is the cells which have been depleted of phagocytic cells, 12 million, no response to sheep erythrocytes. Now we take 3 million of the adherent fraction containing the phagocytic cells. We have a very low immune response, virtually no immune response. If we take 1 million, we have no response. In other words, we have not enough cells here to respond. That was the conditions we wanted to choose. If we now mix lymphocytes, 12 million, with 3 million phagocytic cells, we restore the immune capacity of that suspension. If we take a ratio of 12 million to 1 million phagocytic cells, we can restore immune capacity as well. Now, based on these findings, then we would conclude, if we are allowed to transfer these findings to the situation as it exists in the jugular body, we would conclude that this soluble protein antigen from this bacteria is capable of inducing antibody-forming cells in the jugular bodies. Next slide, please. Here we have enumerated antibody-forming cells at 3, 5, and 7 days. Here in the jugular body of the toad, here in the same animals in the spleen. And you can see that on a per million basis, the jugular bodies are just as good as the spleen. And in fact, the jugular bodies may be superior to the spleen on a per million cell basis. That is, antibody-forming cells per million nucleated cells. But because the spleen has other cells present as well, we have calculated the total number of antibody-forming cells, and there we found that the spleen produces something like 4,000 antibody-forming cells, and the jugular bodies about 600 antibody-forming cells. And if you assume a mean of about 6 jugular bodies per animal, then we can say that the jugular bodies contribute at least as much to the immune response as does the spleen. Now, we were interested from what we found in the fish to look at the morphology of these antibody-forming cells. Next slide, please. It is known to you that the morphology of antibody-forming cells in mammals is a very heterogeneous, gives a very heterogeneous picture. These are the antibody-forming cells, just a random collection of antibody-forming cells from the mouse spleen, small lymphocytes, larger lymphocytes or basophilic mononuclear cells, up to the immunoblasts. Here we have a dividing cell metaphase in chromosomes in the metaphase. That's the mouse. Now let us have a look at the toad. Next slide. We have, I would say, exactly the same picture. We have a very large heterogeneity in the morphology of these cells. And the important point is again the small lymphocyte. We have him present here in the toad as well as it was present in the mouse. We now carried out kinetic studies on these different cell classes. Next slide. In the toad, we have, as we would expect, a total increase in the total number of cells per spleen in the large cell categories, as we would have expected. And we found, using treated thymidine labeling experiments, that up to between 60 and 80% of these larger cells are taking up thymidine. That is a very high rate of thymidine uptake and suggests some degree of synchronous proliferation. This was upon 3 hours in vitro labeling. Then I throw your attention to the small cells. These small cells do never label when treated thymine is given. They appear suddenly and then they slowly disappear again. That is in the toad. Let us have a look in the mouse. Next slide. In the mouse, the same situation for the larger cells, and here the small cells again. They are there at the very beginning of the immune response. That is, at the time we can routinely assay for antibody-forming cells. And upon pulse labeling as well as continuous labeling with tritiated thymine, that is, injecting tritiated thymine at time of antigen injection, and then every 6 hours giving a shot of tritiated thymine, never do we find these small cells being labeled. I think we can conclude from these data that in the toad as well as in the mammal, there are small antibody-forming cells present which appear by differentiation rather than proliferation, whereas other cells, larger cells, certainly contribute to the immune response by proliferation. Now, from the toad, we go a step higher to the reptiles. Next slide, please. We were lucky enough to obtain the most primitive, only a couple, a female and a male, of the most primitive reptile, still existing reptile, the tuatara from New Zealand. We expected to find a lot in these animals. We felt, since they are at the very close to the branching off point, in one direction to the modern reptiles, in the other direction to the birds, we expected to find a bursa-like tissue in these animals, that is the appearance of a second primary lymphoid tissue, and certainly expected to find some jugular body-like secondary lymphoid tissue. But it was a disappointment. In both animals, we could not find any secondary lymphoid tissue apart from the splenic white pulp, and there was no suggestion that there was any gut-associated primary tissue. Now, it may be these animals were too old, and we should have a look at the young animals, but they're so difficult to obtain that I don't think this is possible. The immune response of these animals, next slide, was another disillusion. It took them a tremendously long time to reach quite a high titer of antibody. That was at 18 degrees. You cannot keep these animals at temperatures which vary very much from the temperatures at which they are in their normal biotope. And that's 18 degrees as the mean temperature in their natural environment. And it took them a long time, and when we gave them a second injection, here for the male, here for the female, we did not observe any sign of a secondary anamnestic immune response. From here, let us go again a step higher, from the reptiles to the most primitive mammals. We had the unique opportunity to look at the most primitive mammal, the Echidna, Tachyglossus aculeatus, which belongs to the monotremes. This is an animal which is an egg-laying animal. The egg is put into a pouch, the young hatches there, and is fed on the mother's milk. You can see that in the phylogenetic tree, the Echidna is very close to the therapsid reptiles, and thus really can be regarded as one of the most interesting mammals with respect to its phylogenetic, immunophylogenetic position. Next slide shows us such an Echidna. It has spines, it has a pointed nose. Next slide. And when we performed lymphangiogram in such an animal, we found that there are a very large number of very small nodules, especially in the thoracic, in the thorax region. Here you can see the thoracic duct. The injection was performed in one of the hind legs. Upon biopsy, these little nodules turned out to be lymph nodes. Next slide. But a very particular type of lymph node. If I may have this in focus, please. First, these lymph nodes were inside the lymphatic vessel, usually suspended on a vascular bundle, and they consist of a cortical area consisting of densely packed lymphocyte and a central area which proved in functional tests to be a true germinal center. Now let's see what these lymph nodes do upon injection of radiolabeled antigen. Next slide. The antigen is first taken up in the periphery and then moves towards the center of the node. And when we looked at the electron microscopic pictures, we found exactly the same surface attached antigen as I have shown you to be the case in the toad and as Nossel has shown to be the case in higher mammals. From these studies, we concluded, next slide please, that such a lymph node in the echidna, in the monotremes, one single lymph node is equivalent to one lymphoid follicle in the cortex of a multifollicular lymph node in higher mammals. Next slide please shows this in the previous slide, I'm sorry, was from the rat. This shows it, the same story in a more schematic diagram. Sometimes we could find lymph nodes which looked as if they had fused together, in that they had a sort of a germinal center which had this shape, and other lymph nodes seem to have fused, in which we then found two germinal centers. Of course, we haven't got the sequence of this occurring, but it is at least a suggestive evidence that that may be what happened in evolution. That is, that this lymph node can be regarded as a true analogous to fusion of such single lymphoid follicles to the multifollicular lymph node in higher mammals. Now let us have a look at the last slide, which shows us the immune response exhibited by these monotremes. Here we have the primary immune response. Here we have the immune response after the antibody has been treated with mercaptoethanol. Here we have two animals which showed quite a reasonable degree of an amnestic secondary response. This was, however, an exception. Most of the animals do not exhibit anamnestic responses, but that may depend on ratios of antigen doses between primary and secondary response. And I feel this could be clearly regarded as an anamnestic response with more than 1 log difference between the peaks of the primary and secondary response. This is somewhat less dramatic. Let us then come to the conclusions. I have forgotten to tell you that the monotremes, for the first time in evolution, also show the presence of clearly defined pious patches. They have an appendix, they have tonsils. They look in any respect like higher mammals, except for these lymph nodes. It seems self-evident that there is a correlation between the evolutionary status of a species and the degree of organization with respect to secondary lymphoid tissue. We assume that the selection pressure for the evolution of secondary lymphoid tissue must depend on factors determining the quality of the immune performance. There is good evidence that an increase in the total mass of secondary lymphoid organs also results in an increase in the capacity to retain antigen at locations of strategic importance, such as in the lymphoid follicles. It is suggested that an increase in the complexity of secondary lymphoid tissue organization is paralleled by an increased complexity of the cellular mechanism of the immune response. And finally, this is rather a suggestion, it is obviously time that we try to find correlation at the functional level rather than at the comparative descriptive level between such observations at the made in the field of comparative phylogeny. This requires the increased application of standard experimental techniques at the cellular level into research on immunophylogeny, and I think such techniques are now available. We have methods to identify single antibody-forming cells, to quantitate them. I think it would be of exciting interest to look, for instance, what the hagfish does with respect to cells which cause bacterial adherence. It may be that's the only way how the cellular part, the cellular involvement in these lower species could be studied with respect to antibody formation. Thank you.